Sir, a very good morning, sir.
Good morning.
Sir, firstly, we would like to extend our warm greetings from Santhiram College of Pharmacy, sir, for your likely acceptance to present your eminent work or eminent to share your valuable words in this conference, sir, to share your valuable words. So here, we also welcome all the delegates for your timely response to participate in this Impact Lecture Series 2022-23 organized by Institute Innovation Council on the theme Entrepreneurship and Innovation, Session 2, which was organized by Santhiram College of Pharmacy, Nandyal, in association with MHRD, Ministry of Education, MOE, Innovation Cell, Government of India, and in collaboration with AICTE. We heartily welcome you all. So now let us start our today's webinar in the name of Almighty with the lighting of lamp. Now I request Dr. S.K. Suresh Kumar sir, Santhiram College of Pharmacy to address our today's gathering. Sir, please.
Dear delegates, good morning. So on behalf of Santhiram College of Pharmacy, so I welcome you all for an Impact Lecture Session 2, which is the theme of Entrepreneurship and Innovation. And for the same we have the speaker Dr. Chandraya Godugu, Assistant Professor, Head of the Department of Pharmacology, NIPER, Hyderabad, and he is delivering his topic with the promising trends in the pharmaceutical sciences. And we have another eminent speaker, Mr. L.A. Ramaprasad, Director, JSL Health Sciences Private Limited, Hyderabad, and he is delivering with his topic, the process of establishing a startup. So I hope this information is, the today's session information is very useful, like our previous session 1. So with the permission from Dr. Chandraya Godugu, so we are going to session. Sir, morning sir, Chandraya sir.
Good morning, sir.
Welcome sir. So we can start our session, sir.
Yes, yeah. Thank you.
Sir, thank you very much, sir, for your addressing, sir. With no delay, let us start our today's Impact Session 2 by inviting our speaker, Dr. Chandraya Garu, Assistant Professor, Department of Pharmacology, NIPER, Hyderabad. Now, I would like to ask Maheshwar Reddy Garu to give a brief biodata about Chandraya Garu. Sir, please. Maheshwar, sir.
Thank you, sir. Good morning to one and all. This is D. Maheshwar Reddy, Associate Professor, Department of Pharmacy, Santhiram College of Pharmacy. It is a great pleasure for me to introduce our speaker today, who is going to talk to us about the promising trends in pharmaceutical sciences. His name is Dr. Chandraya Godugu. Coming to the academic and professional details, Sir completed B.Pharmacy in 2003 from UCPSC Cocktail Institute. Got gate rank in 2004, scored 99.55 percentile with all India rank 39. Sir completed M.Pharmacy Pharmacology and Toxicology in 2006 from NIPER, Mohali. Later, he received PhD award in 2011 from NIPER, Mohali. Sir completed postdoctorate from the Florida and University in 2003. Sir having nearly 11 years experience in research and teaching. Sir now working as an assistant professor in NIPER, Hyderabad. Sir guided 55 M.Pharmacy dissertations and 10 PhD students under his guidance. Coming to the publications, Sir published more than 140 publications in national, international journals with a high impact factor. Sir given more than 80 poster presentations and 28 invited talks. Coming to the funding projects, Sir received grants more than 2.5 crores rupees from the Department of AICTE, DST and DBT. Last but not least, Sir received more number of awards in his career from various films. Thank you, sir.
Thank you very much, Mahesh Reddy garu. So I will start now, right? So at the outset, let me thank all the organizers, Principal Dr. Suresh Kumar sir, and all the faculty who are actually joined today. And my special thanks to Shivashankar Reddy for inviting me for this important lecture series, which is Impact Lecture, and, supported by AICTE, where different institutes are joining to understand what are the different innovations happening in the pharmaceutical sciences. And also, I would like to all the participants who have joined from different institutes. Despite of Saturday, I think I could see many people joined here. More than 41 people. I'm sure many of the people are also joined through the YouTube. So actually, as I mentioned, my background is already clear. Actually, I'm a Niperite from my master's onwards, and I did my postdoctoral. And I came back to India in 2014 and joined as Niper Hyderabad as assistant professor. I have been continuing it. It's been a wonderful journey for me in Niper Hyderabad during my studies time in Niper Mohali also. So I'm a proud Niperite, and I would like to represent how Nipers are doing research in different disciplines. So I'm actually a pharmacology and toxicologist. I actually teach regulatory toxicology. I also work with nanotechnologies. I have 3 branches combined now. So I have pharmacology as a education background, and I teach regulatory toxicology, more of a toxicology subject. I also work in the area of nanotechnology, nanotoxicology. That's the brief about me. And let me share my presentation, then we'll go into presentation. So if you have any questions, actually you can just type there, then at the end I think we will be able to discuss better. In the online mode, if you have a question, I think stopping in between, I think it's not a good idea. I think let's have the questions and answer session at the end. I'll request organizers to note down the questions so that we can discuss in detail at the end of the session. Right, sir? So let me share my presentation. I hope I'm clear. My voice is clear to you all, right? My voice is clear to you all?
Yes, sir.
Sir, it is clear, sir.
Great. Okay. Let me just share my PPT. Is it move to next slide? Which slide we are on right now?
First only, sir.
Is it not moving to second slide?
No.
No, sir.
No movement?
No movement.
No moments.
Is the issue I'm facing?
Yes, sir. Now we can see, sir.
But the problem is this is not in presentation mode,
Yes.
But is this visible to you people? Do you think we can able to?
Yes, sir. Yes, sir. We can read, sir.
Are you sure? Because I am going to show a lot of figures. We may have a problem there. That's what my worry is. Okay. When I increase this,
Sir, can you maximize, sir, in the slide share mode? Is it
that's what
possible?
I'm. Okay. What do you want me to do?
Sir, in your sir, from your side itself, can you go with the slide share mode, sir?
I'm already doing that actually.
Okay,
I'm already doing that.
sir.
That's where I'm again trying to. Should I reshare again? Should I become a presenter? Maybe it might allow me to. It's not sharing, right?
Get shared, sir.
It's not okay. How is this now? Are we going to the next slide?
Yes, sir.
Are we going to the second slide and all slides?
Yes, sir. Yes, sir. Yes, sir.
On the first slide now, right?
Yes, sir.
Thank you. Always a problem when we have to share it. Sometimes it won't share the full screen, and sometimes next slide won't be. I can see the next slide moving, but we are not able to see it. So that's always I have a concern when I just started. So sorry for the trouble, and let me just quickly jump into the presentation. I just kept the title "Promising Trends in Pharmaceutical Sciences." If we take the promising trends in pharmaceutical sciences, it can be huge, right? We can just take one subject area and you can talk about what are the different promising trends. If you more deeper you go into the more interesting it will be. We can take any example and go for. But I just want to go peripherally. What are some of the promising trends which are happening in the pharmaceutical sciences, where we can actually, some of us, including me, can pick up some of the those trends to read more, or also to start working in this areas based on the facilities available with you, or based on the infrastructure present in the institute, right? So I mentioned it's my title, and our department is a department of biological sciences. Why we call biological sciences is biological sciences has 3 departments now. I'm the head of the department of this department. We have pharmacology and toxicology. We have a regulatory toxicology. We also have informatics, pharmacoinformatics. So these 3 actually come in the drug discovery and development. That's why these all the 3 subjects are talking about biology. That's why we made them into biological sciences. So ours is a big department in Hyderabad. We have 6 faculty, and we all work in different areas, including infectious diseases, inflammatory diseases, several CNS diseases, and so on, right? So my area, my research area is more into fibrosis, cancer, and several other inflammatory disease conditions. So what we talk about is, initially we talk about the drug discovery development, and we see how the cancer is treated and where we are facing a problem with the cancer, and what are the challenges we are facing with the cancer. Can we actually troubleshoot them by using some of the tools like molecular technology, or targeted therapy and targeted therapy. And we also talk about some of the siRNA, shRNA, microRNA based therapeutics. Then we also talk about monoclonal antibodies. We just briefly cover about the CRISPR-Cas technology. We also talk about CAR therapy. These are all the promising trends I feel, and one should know in the current generation. So because these are the fast-moving areas where drugs have already started coming into the market based on this. So that's the background of it. And we can see there. Can I ask others to mute? There's a lot of disturbance. I request all of you to please kindly mute, because I see a lot of echo happening here. So drug discovery and development at a glance, and I don't want to bore you people, and I just want to mention here is it's a very long-term process, right? And almost more than 10 years time it takes. And why it takes a lot of time is it's not at the discovery level, it is the development level. The amount of the preclinical studies we have to do to prove safety, amount of the clinical trials we have to do to demonstrate its efficacy and safety needs a lot of time. Do we have that much time? 10 years of time if we don't have a drug in that particular category? We have so many orphan diseases where we don't have any drug for particular orphan disease. Orphan disease means where the number of the patients suffering from that particular disease condition is very limited, less than a certain number. Also, we don't have any drug in particular conditions. That's why they call orphan diseases. So, are any other disease, even including the COVID? We never had a drug for COVID, right? Though we tried different drugs, they're all actually not very scientifically proven. We have used remdesivir, we have used chloroquine, hydroxychloroquine, and none of them actually really demonstrated proven 100% efficacy. Like if you take a painkiller, you are sure that the pain will be reduced, more or less. And if you have a fever, you are sure that if you take one fever tablet, it will be reduced, normalized. So that's what is the medicine is working. But that didn't happen with the COVID, right? When we had a COVID, though we tried different drugs, but I think our body only recovered from the infection over a period of time. That means such kind of conditions like COVID, we cannot wait for 10 to 15 years. Of course, there are expedited drug discovery and development process, or you have to only repurpose the drugs which are already being available in the clinical use, right? And I hope you all know what are the advantages of the repurposing of your drug, and there is a lot of scope for us to repurpose the drugs. Metformin, we all know that it's an anti-diabetic drug, but if you just search in the PubMed, metformin and its for other indications, it's been explored everywhere. It's a AMPK activated, it's artificial inducer, and so on. It has been very hugely explored for different disease conditions. The beauty with metformin is well proven. Well proven, why I'm saying is because the person can take a drug for a long time, and over once they have it's been diagnosed with diabetes, and the person may be taking the medicine for very long years, maybe 10 years, 20 years, and even lifelong, right? So if any drug which is found to be safe by taking the drug for over a period of time, several years or several decades, the safety is already well proven. That means such kind of drugs, if we can able to repurpose, that will be a wonderful strategy, right? Suppose if we have a condition where you think metformin may be a good candidate to actually treat that condition, you should right away explore on that. The reason is of the safety of the metformin, right? And if we since safety of the metformin, as well as they no need to do any preclinical trials again, we have to only do some of the clinical trials in a selected people. Why you have to do clinical trials? Just you have to do that, yes, in this particular condition also, this particular metformin. If I recently come across a paper in Nature Medicine where metformin is reversing the fibrosis. Pulmonary fibrosis is one of the very serious condition. It's been increased post-COVID complications for pulmonary fibrosis, where the lung is becoming a scarring of the tissue in the lungs. So such kind of disease conditions where we have only two drugs, if metformin can able to reverse it, it's very good, right? Because generally it's believed that fibrotic diseases are not reversible. But the Nature Medicine paper, it's not the ordinary paper. Nature Medicine paper is talking about metformin is reversing the pulmonary fibrosis. So that's where the repurposing opportunities are right. So you see the cost of the discovery development the process, almost 2 billion USD. That means almost 16,000 crores of rupees are required for us to develop a drug. So it's a lot of money. That's why small companies will not able to do it, and they only develop up to certain level, up to certain stage. They just design the molecule and they do the in vitro screening, and they establish the proof of concept. From there onwards, it will be going to the next larger organization like Pfizer or the Johnson & Johnson. Know what is they will be take over that particular molecule, and they will take the risk of developing. Why I will say it's risky is the molecule is failing at the clinical level. It's going to be very costly failing, right? That's why in drug discovery and development, early killing is the best strategy. That means if you want to kill the molecules in the early stages itself. Don't wait the molecule to be killed at the clinical stage 11, phase 1 or phase 2 or phase 3 times. If the molecule fails at the any of the later the phase of the clinical trial is more costly, the loss is going to be great. Example was Dr. Reddy's has also suffered a failure in the clinical trial with I think Balaglitazone, which was anti-diabetic drug, which was under the development process during 2004 or 2005, and it was observed that the compound was causing some kind of cancer. So it's causing a cancerogenesis in some of the side effects. By the way, you have to make sure that when you develop the drug, you have to generate a 2 years exposure data to the chemical, and you say that yes, my compound is not causing any cancer. There's a this toxicity study called 2 years by essay, called the carcinogenicity essay. You have to give the compound for 2 years, either orally only, or through the water, or you have to add to the feed. After 2 years of exposure, you have to open the animal and you have to understand all the organs, how the organs are there any cancer, are there any lesions developed in the any organs needs to be established, and we will be comparing with the control animals. If the number of lesions are increased in the any of the treatment group, then probably you may have to think one more time whether molecule can go into clinical trial or not, because and it takes 2 years time, right? That means for a molecule to go to clinical trial, you need to show that it's not carcinogenic. What I say is right. Well, will you depend on the 2 years per se, or will you depend on the shorter per se? There is a lot of studies happening in the toxicology area as well. Okay. That means drug discovery and development is going to be very costly, right? It's time as well as the cost involved in that, and the kind of impact you can create if you are failing it. That's why those small companies are doing a lot of discovery. Development process will be handed over to larger organizations. Larger organizations are able to handle this particular loss because they have multiple sources. They just don't depend on one molecule. They have a multiple blockbuster drugs, or the top selling drugs, or the top innovation products. So they are okay to. That if they're failing in one of the molecules also, they're still able to survive. That's how large companies always do it. And we all know that where from the new drug can come. Mostly earlier the new drugs are coming from natural sources, natural products, or chemical based ones. Nowadays the source has been changed now. Now we are talking more of than biologics, right? We are talking the biologics and biosimilars. Biologics and biosimilars are more of the biological products. They are more specific to disease, so we can expect less safety concerns. However, they are going to be very costly. Biologics are going to be very costly. Example, insulin. Of course, it was costly earlier. Now, because of the RDN, recombinant DNA technology, the insulin cost has been decreased. Otherwise, if you take any tocilizumab or any other drug which we are trying to use during COVID complications, was very, very costly, right? 40,000 rupees, 50,000 rupees, just injection. So all the biologics are very costly, and that too during the first 20 years of the innovation, they are going to be very costly, because the company we have discovered that particular biologics going to charge high, and most of the times these biologics come to India later, not immediately after they discover. That's under patent, right? So if you see the drug discovery document, we have target validation where you have to start with 10,000 molecules to 20,000 molecules. Just imagine 10,000 to 20,000 molecules. Big number of the molecules you have to design. I have a screen in the computer software tools or the computer aided tools to identify which molecule is going to bind to the right target, and which one is going to have a good biopharmaceutics and pharmacokinetic properties, and which molecule is also going to have a safety profile, better safety profile. So you have to see all this also. Target validation is not about the safety profile. Target validation is more of which is the molecule better fitting it. We have 5-6 different receptors of the different types. If your drug is your disease is only specific to certain receptor subtype, your target your new chemical entity should be only binding to the particular subcategory of the receptor, or the subtype of the receptor. Then only you can expect a better safety profiles, right? So target validation, you are starting with the 10,000 molecules. Lead optimization, once you identify some of the molecules are better binding to the target, then you can take 50 molecules to further optimize the lead. Why do you have to do the lead optimization? Lead optimization is required because probably the drug needs to be given orally, because for long-term treatment it has to be orally. You have to see that biopharmaceutics proper solubility and hydrophilicity, lipidicity are proper. In Lipinski rules needs to be kept in mind here, as well as the stability of the molecule and any reactive groups present in the molecule which may be causing a toxicity. Can they become accumulate in the body? They're accumulating in the body, they can also cause a toxicity. So you have to do a lot of optimization there. Once you've done the lead optimizations, 2 to 5 molecules will be sent for the preclinical testing. Preclinical testing in the sense in the animal models. What you do in the animal models, you try to understand in the particular disease condition, whether it's a diabetes or hypertension or cancer, you will be developing the model in the animals, and you see how this particular 2 to 5 molecule which you have optimized based on the filtering strategies whether they are actually anti-diabetic or not, you will be understanding it. Once you understand the anti-diabetic property or anti-cancer property of the total molecule, now we'll be doing a lot of safety studies. How do you do the safety studies? If you see the ICH guidelines, International Council on Harmonization, QSEM, quality, safety, efficacy, and multidisciplinary. So in that out of QSEM, safety guidelines are very important. If you just go out to the ICH guideline, if you just search the safety guidelines, S1 to S11, there are different types of guidelines. What type of toxicity is to be done, and what model to be used, how many number of animals to be used, everything will be given. That means when you do the preclinical studies, the amount of the animals used in the toxic studies are much higher than the pharmacological studies. Pharmacological studies, maybe you'll be needing maybe few hundred of animals to prove it's effective or not. Whereas in the toxicity can be happening from different types, right? It can be genotoxicity, it can be carcinogenicity, it can be reproductive toxicity like thalidomide condition, it can be phototoxicity, it can be local toxic, it can be a organ-specific toxic, like that. You may have different types of
Hello, sir.
toxicity concerns. It can be phototoxic, it can be a skin sensitizer, so on. Like it can be a tubular prolongation, torsade depointase. We all know torsade depointase, right? Terfenadine was the drug which was withdrawn from the market after clinical usage because it was causing a sudden cardiac deaths. Sudden cardiac deaths were happening because of tubular prolongation. Because it was extending the QT interval of the ECG that was causing the ventricular tachycardia. It's called tubular prolongation. So that's ICH S7 guideline talks about the tubular prolongation. So that means you have to do a lot of studies in the animal models and in vitro models. Once you are sure that all the studies are proving that yes, my molecule which I have optimized based on the target validation, lead optimization, found to be safe, and it's not posing any risk to the animals. I'm expecting the same profile might be also happening in the humans. Based on the data, once you give the data, we can go for the clinical trials. That's where you will be doing the IND, investigation application. So investigation application will be having all the safety and animal data, including the chemical properties and all, will be submitted. And based on that safety profile, you'll be given the chance to plan phase 1 or phase 2 or phase 3 clinical trial. Of course, phase 4 clinical trial also will be there. And each stage will be depending on what kind of animal toxicity data is required. For phase 1 trial, you may not be needing every detailed toxicity data, because phase 1 trial is only given single dose. Exposure is going to be very limited, and you're not going very high doses. It's small dose you are starting, right? This will be more of the tolerability as well as the pharmacokinetics will be understanding it. In phase 2, you'll be only studying in a group of 100 people or 200 people for its efficacy. And the moment you go to next phase level, the number of patients, the centers may be increased to understand the efficacy as well as the safety of the molecules will be understood. So again, another way how you do the drug discovery process, we have to do this proper target selection is required. Then the lead discovery. Then medicinal chemists have a lot of role to play here, because without medicinal chemistry, we're not able to get the new drug. Earlier, we are fortunate enough to identify the drugs from the natural sources. But it's like, how do you know that particular plant molecule may be having a certain activity? Of course, we have an ancient history that this plant were used for a certain condition, but they are not scientifically proven, right? Of course, there might have been evidences directly exposing to the humans, but nowadays we cannot take that particular approach, right? We need to be very scientifically sound that for any molecule to get approval from the regulatory bodies, you need to make sure that its mechanism, its safety, and its fate of the molecule, everything needs to be understood. So medicinal chemistry is very important, and people nowadays complain that we don't have a proper medicinal chemist. We don't have a good medicinal chemist in this country to lead the drug discovery and development. That means there's still a lot of opportunities in the medicinal chemistry where you actually integrate the chemistry knowledge with all the targets. Docking studies are in silico docking studies to the molecular modeling tools, as well as nowadays people are using artificial intelligence and machine learning. How that is useful? Because artificial intelligence and machine learning can able to better design the molecule for you because it handles huge amount of the data. It might tell you, look, you don't need to just start with the 10,000 molecules. So I'm giving a certain filtering strategy. I'm giving a certain my own calculations of artificial intelligence and machine learning, and I say that if you only take these 100 molecules, not 10,000 molecules, just only concentrate on these 100 molecules. I have designed these 100 molecules based on the filter criteria I have given. It will tell that these are best fit molecules. Now, medicinal chemistry job may be easier now, right? Because we have used artificial intelligence to design the molecules, and they actually the better prediction can be expected. So then just synthesize those 10,000 molecules. Then easy for the chemist to synthesize those molecules, and also easy for the pharmacologist or the biologist to screen those molecules. Rather screen 10,000 molecules, if I only get 100 molecules, I can better focus on them, right? How they're actually behaving. It 10,000 molecules, immediately we have a lot of high throughput screening, where you have to only use single concentration and then two concentration. Whereas we have limited number of compounds, wide range of concentration can be studied, and that's where you'll have a more flexible. So you will be always doing in vitro studies. In vitro studies in the sense, not necessarily it has to be cell line, it can be also enzyme-based assays. The different kits are there. For every target, now the kits are available. If you have any target like topoisomerase or any enzyme for that reason, we have kits available. You just have to purchase the kits and do the simple procedure given in the protocol, and you can able to identify whether the molecules are binding to that or not. The initial step should be whether your target is hit by your molecule or not. The 100 molecules or 10,000 molecules, what we have. So based on the molecules which are properly hitting the target, or the binding to the receptor, or the protein, or the enzyme, then we will be selecting the best few molecules to go for in vivo studies. That way you do in detailed studies and so on, right? So in this process, you can actually always use omics. What's omics? Omics is omics itself is a large number of the markers studied together. Omics, large number of molecules studied together. What molecules we are talking about? Molecular patterns studies together. What molecular patterns we are talking about? It can be proteins, it can be genes, it can be metabolomics, it can be lipidomics. What is lipidomics? Lipidomics is not about the human body lipid profile. Lipidomics is more of a how cellular lipids. Cell can contain large number of lipids, ceramides, cholesterol, phosphodiester, and so on, and sterols. These all the lipids have a certain role, and it's found that certain lipids are also found to be altered in certain diseases. So you can also use the lipidomics, is the omics too nowadays. People are using it. It's a metabolomics where you understand how the cellular metabolism is altered. Cellular metabolism, glycolysis, fatty acid oxidation, and mitochondrial respiration. How these different metabolites, Krebs cycle, the relevant metabolites, how they are formed, how they are altered in disease condition, is the metabolomics. And genomics means how the messenger RNA is altered in a particular toxic condition or the disease condition. So anytime if you don't have any idea about the target, what tools people are using is omics tools or proteomics. Proteomics is what you understand just an entire protein profile of the cell or the tissue. Can you imagine how many proteins may be there in the tissue? It can be 10,000 to 20,000 proteins may be there in the tissue. If you take just single cell, the number of proteins may be 10,000 to 20,000 or more than that even. This many number of proteins are there in a cell. Now we will be trying to understand all the proteins, how they are actually altered in the disease condition. This is called omics. If you don't have the omics, what will you be doing? You will be doing only trying to understand only one protein through the certain ELISA based technique, or a certain Western blot technique, or some other immunohistochemistry based technique. These are the techniques to understand one protein. If I have no idea what protein is altering out of 10,000 proteins, the best strategy should be profiling entire 10,000 proteins, or entire the cellular proteins present in the tissue or the cell, and compare the normal tissue with the diseased tissue. other toxic, treatment tissue. If you can able to see the pattern, what type of proteins are altered, now you can narrow down from 10,000, maybe you can narrow down to some hundreds of proteins. Now, these proteins also now found to be linked to the certain pathways, right? This particular protein is involved in the G protein coupled receptor signaling. Another protein is involved in the mitochondrial ATP generation. Maybe a different protein may be involved in the ion channel functioning, or other proteins may be involved in the enzymes. Like that, if you know the particular protein, you know the function of the protein. That's called protein annotation. Based on that, you will tell that, okay, these proteins are playing a role, and this is my new target I have identified based on my omics tools. This is how the new target can be identified by using the omics. If you take the clinical samples, you can always get right when people get the biopsy done. You don't need grams of tissue here; just need a milligrams of tissue to do the proteomics or genomics. So proteomics studying the protein, genomics studying the messenger RNA. So what is the link between the messenger RNA the protein? What are the amount of the messenger RNA expressed? Correspondingly, post-translational, you will be seeing a protein formation. Generally, the more messenger RNA, more protein can be expected. But you have to understand what is happening at the messenger level, mRNA level, or at the protein level, you will be able to understand. That's how omics is a very important tool. Of course, it's not everybody's field nowadays, because you need a sophisticated tools to handle this area. Sample preparation is very costly. Sample preparation itself needs a lot of equipments, and you have to handle a very minute volumes, not the mls. You have to handle few microliters of volumes. In that, you are going to subject your samples to mass spectroscopy or any other tool, which will be giving you what type of proteins are there. So a lot of optimizations are required, but this is the tool people are using it to understand the new targets. Not only new targets, also the safety profile. Why I'm saying safety profile is when you give now you are sure that I have identified a very good molecule, and it's working very well for the diabetes. It's working very well for the COVID. Now, safety wise, we are only doing animal studies. But animal studies, we give the compound to the animal. What you do, you take the biochemical profile, you take the organoids, you take the blood profile, you also take the animal body weights and that. These are the only things, right? But not necessarily that animal is going to lose the body weight. Animal is directly going to get affected at the organ. There may be no change in the blood. There may be no change in the biochemical profile, because the body has a lot of compensating mechanisms. But you are missing some of the toxic profiles which might be happening at the molecular level, at the cellular level, the protein level, gene level. So if you use the omics for the safety evaluation, I think you can able to better predict the safety. That's why omics tools not required by the regulatory agencies to do the safety evaluation, but a company which is well equipped with the omics tools, they may be subjecting the promising molecule in the omics to understand whether whether the molecule is going to be causing any perturbations in the molecular events or not. If that is causing any alteration in the disturbance in those events, probably they can focus on that. Look, this particular molecule is going to inhibit particular certain protein, which may be vital for your cell to function. There may be a potential toxicity can happen. So you can actually better predict it. That's how by using omics tools, you can able to kill the molecules early. If you kill the molecules early, so you can save the time, as well as you can also save the money, as well as you can plan the next better molecule than the molecule which you are trying to work on. So these are some of the aspects people have been working on it. It's every field, every topic is a book. If you just take the metabolomics, there is a book available. If you take proteomics, a book. A lot of literature available. The way you prepare the sample, the way you analyze the sample, everything is very deeply covered, and if anybody is keen, should actually read the relevant literature. I suggest, right? So let me just quickly jump to cancer. Why we have to think cancer is? I think this is a lot of research has been done in this area, but still also there is a lot of unmet medical needs. We don't have very good chemotherapy available. What are the chemotherapy you are taking? I don't think so any chemotherapy is effective. If you just recollect also, generally what happens is person takes chemotherapy and within 1 year or 2 years, you just generally normal hear that the person is no more. And it's strongly believed that it's not the cancer which is killing the patient, the cancer chemotherapy which is killing the patient. It is true only because cancer, what it's causing, it's trying to occupy the normal healthy tissue with the malignant tissue, and also it's spread to other parts of the body. It takes time, right? Whereas in the chemotherapy, radiation therapy, strong chemotherapy like doxorubicin, the cisplatin, Taxols, what they do is, they actually brutally attack your body. There is no discrimination between the healthy cells and the malignant cells. They actually attack all the fast dividing cells like bone marrow, the epithelial lining, the G80 and the lung. That's why you can see a lot of toxicity issues. And I have not had any case where person takes the chemotherapy and never complains. We are always going to be a lot of complaint on the safety concern. That's why we still have not met. Yes, here also in this field, there's a lot of targeted therapy happening. Targeted therapy means you actually identifying what is the receptor expressing that particular tumor. Then you try to develop you have developed the specific therapy against that, like monoclonal antibodies like trastuzumab or Herceptin for the breast cancer, and we are actually using that. Still also, we are not successful to completely eradicate it. There are certain reasons for that as well. So if you just see the characteristics of the cancer cell, and self-sufficiency in growth signals, it don't need everything. It's kind of more adjustive in nature. If whatever I have it, I'm okay, I'm ready to survive. I don't wait for all the things to come to me to survive. That's how self-sufficiency. What I have it, I think I'm sufficient to survive today. When I have a good resource, I will enjoy that. If I don't have also, I'm still okay to survive self-sufficiency. That's there. Evading apoptosis. Apoptosis, the programmed cell death, and cancer cells all avoid this apoptosis. Programmed cell death, your body has a very good apoptosis because tumor suppressor gene is active in this particular condition. And what happens is all the cancer cells evade the apoptosis. They escape from the apoptosis. So that cell death is already reduced in the tumors. That's how your body, your immune system is also compromised in the tumor. So immune system is also not active. Insensitive to anti-growth signals. Why anti-growth signals? Why your brain or your liver or any tissue is not going to the level they should go? Why only your liver regenerate to certain level and stop? Because you have anti-growth signals. If your body has your tissue, your cell has a signals. Okay, the required growth has been already achieved. Then it will send the signal that growth needs to be stopped. That will be happening in the healthy organs, whereas the anti-growth signals will be blocked in the cancer. Sustained angiogenesis. Why sustained angiogenesis is required? Because tumor tissue is not a planned organ. Right? It's just a guest organ happening in your body, in a foreign organ happening in your body. That's why there are no different blood vessels. That means your tissue has to develop the blood vessel, new blood vessel. The tumor tissue has to develop the new blood vessel so that it will get enough nutrients and enough oxygen to survive. And of course, if the oxygen is not there, also still tumor cells are able to survive because that's for the hypoxic condition. Also, they grow better. Due to hypoxia, again it will develop another kind of pathological condition. So again, every environment which is created in the tumor cells and the cancer cells exploit that and try to use in their favor. So sustained angiogenesis is there in the tumor cells so that you will get a good blood supply. And tissue invasion and metastasis, another important one, right? What are the main organs? Generally, you see what are the organs are highly perfused, like liver, your lung, and also the brain. These are the organs generally you see the metastasis happening in. Maybe the tumor might have formed somewhere in the colon, it might have formed somewhere in the breast. What happens is these tumors also migrate to other organs called the invasion and metastasis because they have the capacity to go through the narrow down, they escape from the blood vessel and they can extravariation, intravariation, they all can have it. Through the blood circulation, they can go to other organs and they settle there. One tumor cell is enough to initiate as a foci that will be slowly growing as a nodules. These nodules become a big tumors. That means they have a capacity to go to multiple sites. Once they go to multiple sites, so now we cannot remove all the organs, right? If in the preliminary stage only one organ, like in the breast, the breast condition can be removed. If it's already moved to some other area, how can you really able to remove all the organs. Not possible, right? So this is another concern. Limitless replicative potential. All the tumor cells grow very fast. They have no limitation. So how do you treat cancer? So many targets, so many strategies we have followed. These are some of the strategies we are talking about. We are talking about here. EGF receptor inhibitors, cyclin-dependent kinase inhibitors, which are involved in the growth, immune activating anti-CTLA4 monoclonal antibodies, because it is believed that immune system is also compromised in the cancer patients. So if you can somehow stimulate the immune system, so your immune system is able to attack the malignant cells, because malignant cells are not the healthy cells, right? So if you can stimulate the immune system, telomerase inhibitors, because telomerase are actually the longevity the mediators. So if you can actually able to immortality can be removed. Selective anti-inflammatory drugs. So in COX-2, people have tried to use as a COX-2 inhibitor. People have tried to use as a anti-cancer drug like celecoxib. People have done a lot of clinical trials. Let me take a break. Inhibits the vascular endothelial growth factor, because VEGF is involved in the new blood vessel formation, can be targeted. PARP inhibitors to promote the apoptosis, the pro-apoptotic mimetics, aerobic glycolysis inhibitor, and so on. These are the few, and there are so many other targets as well. So you can actually take one of the strategy and develop the therapeutic. A therapeutic can be a natural product, a plant-based one. It can be a synthetic chemical. It can be also a biological product. Right. Hope I'm moving to the next slide, right? Am I on the nanotechnology slide now? Can somebody confirm this?
Yes, sir.
Great. So, am I slow?
No, sir.
Any suggestion you want me to go slow or fast? Any suggestion you want to do? Any more change required? Going fine.
Sir, you are going fine, sir.
Fine, great, okay. Okay. So now I just want to quickly go to nanotechnology, because whenever you talk about cancer, we have to talk about nanotechnology, nanomedicine. Nanotechnology where you make the particles in the nanometer size. They have certain advantages, not certain, lot of advantages, and also few disadvantages, like in the form of nanotoxicology. Whenever you use the nanotechnology in the medicine, you call the nanomedicine. Mostly for the cancer, people are trying. And also recently, if you see the amphotericin B is used for the black fungus. Black fungus is the post-COVID complication. After COVID, when people have used a lot of steroids, your steroids are immune suppressors. The body becomes so weak in the weakly immune. So whatever the fungus which is there in the plants and the environment, they have attacked the suppressive people, and black fungus enter into the body, and all the teeth, it has also come to the brain, and so on. Right? Mostly the orbital region, eye region, as well as the jaw region got fungus, black fungus got it. And only the drug which was effective was amphotericin B. Why I'm talking amphotericin B is antifungal drug. It's very good. What is the problem with the antifungal drug? Amphotericin B, it is a highly nephrotoxic. More than 70% of the people will develop the acute kidney injury. It can be fatal also. People can maybe also dying because acute kidney injury. Acute kidney injury is a very severe condition, and it was induced by amphotericin B. We are using a drug which is 70% sure that it's going to cause a kidney injury, kidney damage. Such is the situation is. And luckily, there were the liposomal formulation based on the nanotechnology called the ambisome, which was found to be better effective, as well as reduce the nephrotoxicity very, very significantly. So this is thanks to nanotechnology, right? So unfortunately, when the COVID cases have increased in our state, mainly Gandhi Hospital and all, they don't have the supply to develop give to this ambisome. And the cost of the ambisome, if the amphotericin B injection is 250 rupees injection, the ambisome injection was 5,000 or 10,000 rupees injection. That is the difference between the free drug and the nano formulation. There is a technology which is improving the patient outcomes. However, the cost is very high, and the black fungus cases were treated by the medical, all the government hospitals. Where is the government is in the position to get such kind of costly medicines? If the government is ready to pay, where is the supply? We never had a supply. So these are the concerns. So nanotechnology, though everyone talks about it, we don't have good scale of technology where we can actually able to produce it. Like doxorubicin nanoparticles are called the doxil. Why doxorubicin has to be made into nanoformulations? Because doxorubicin produces cardiotoxicity. Everybody who takes the doxorubicin for the cancer therapy will develop the cardiotoxicity, heart toxicity. Now, doxil is the liposome formulation of the doxorubicin, was found to be much safer with respect to cardiotoxicity. At least we are ensuring the safety is taken care of. As I mentioned earlier, all the chemotherapy drugs have a lot of toxicity concerns. If any technology is there which is reducing the toxicity, is great. So that's one area. So nanotechnology and nanomedicine, everybody talks about it, but there is a lot of scope still to explore here. If any members we have who are present in this particular today's meeting need not to be a pharmaceutics person always. A pharmacology person, a medical chemist person can also think in a area of the nanotechnology. A combination, a collaborative work can result in a good proof of concept where you have developed a nano formulation, the liposome formulation, whatever the nano formulation you can develop it, translated clinically, clinically translatable can be seen here. How nanomedicine can actually address the problems we are seeing with the conventional chemotherapeutics. A lot of applications everywhere. You can improve the PK, you can improve the PD, you can do the targeted delivery, you can improve the tolerance of the drug, like amphotericin B. You can reduce the side effects. You can also do the molecular therapy, gene delivery, like a gene delivery as a vaccine. COVID vaccine of this Moderna was actually the nanotechnology driven one, right? They are actually developing the mRNA through through a lipoplexus, which are nanotechnology again. Theranostic application. What is theranostic? Theranostic therapy plus diagnosis. You treat the condition as well as you diagnose how much is it's happening also. Typically, how the nanoparticle design takes place is you have to take a drug. You also have a polymer. A polymer can be a lipid. It can be a PLG or any polymer which is found to be effective, which is found to be safer. You cannot use a polymer which is used in the plastic industry, right? You have to use a polymer which is approved by the US FDA. And also grass polymer, GRAS polymer. GRAS means generally regarded as safe polymer. You have to take the grass polymer which is already used by US FDA for tablet preparation or the injection preparation. You have to select such kind of polymer like polycaprolactone or sodium CMC or the polylactide co-glycolic acid. Like that, you have to take the polymer which are actually safer and they don't produce toxic byproducts when they degrade, and they should be biodegradable in the body. So basically, you take the polymer, you take the drug, and you use different techniques to develop the nanoformulation. It can be a simple emulsion technique, it can be a respiration technique, it can be a size reduction technique. So different techniques are available. For every nanoparticle which you think of developing it, just search in the literature, you find a procedure. Different procedures may be there. Which is a procedure you think is feasible to you should adopt that procedure. The simple one, you should always adopt it. Liposomes are very simple to prepare, You just have to have, what is that, rota operator, To make an liposome preparation, you just need to have a rota operator. You have to take all the lipids, cholesterol and all, egg lecithin and all in the right composition. And you have to dissolve the cholesterol and all the lipids in a suitable organic solvent. Mix the drug also to that particular component, and that all the contents which are dissolved in the organic solvent will be rota operated in the round bottom flask. When you do that, what happens is you are making drug properly dispersed in the lipids, right? Drug is properly dispersed in the lipid, as well as your organic solvent is dispersed. When the organic solvent is dispersing it, the layer is formed on the inside wall of the round bottom glass. Now this the layer which is formed in the round bottom glass can be hydrated. How do you do it? Just have to mix with the right aqueous buffer or the phosphate buffer, and you just have to do the proper mix. Then the layer will be slowly coming into the aqueous in the liquid. Now these other liquids are so simple to prepare, but a lot of optimizations are required. How much drug to be taken? What is the right composition? How much drug has gone inside? What is the loading capacity? What is the size of the liposomes? And the things are actually not so simple, but as a principle simple. But you need to optimize so many things. These are the aspects of pharmaceutics the people. But a pharmacologist, a medicinal chemist also can always think in that direction. Not necessarily it has to be only pharmaceutics area. Of course, they are the pioneers in that. A biologist can also propose it, which like the way I'm doing it. Once you make a nano formulation, it can be liposomes or it can be any polymeric nanoparticle. You can always surface conjugate, you can surface modify. Why you have to surface modify? You have to modify to increase the stability of the nano formulation, like pegylation to improve the stability. You can also add certain ligands on the nanoparticle surface so that it will be driven by the ligand which is present on the surface, like targeted delivery. It can be one ligand, it can be multiple ligands. So multiple ligands will be driving your nano formulation to go to the right ligand which is having a receptor present on that particular tissue. If it's a cancer, you can take the cancer receptor as a targeting strategy, right? So cancer targeted delivery through nanotechnology is very common. Inside the nano formulation, inside the body of the nano formulation, you can have a drug. You can also have a diagnostic agent. You can also have the magnetic contrasting agent. You can also have a another agent. So you can use the diagnosis as well as the therapy, right? So if you see the cancer targeting, it can be passive targeting, active targeting, as well as the trigger release targeting. What is passive targeting? If you see the tumor is having a leaky vasculature. Why it's a leaky vasculature? It's leaky vasculature because tumor endothelium is kind of having a gaps. Why the gaps are there? That's why it's leaky. So whenever you give any particulate form, particulate form, it will be actually easily entering the tumor tissue because the leaky vasculature. That means it's a passive targeting. You don't need to have any surface having any surface decoration with the ligand. Just have to make a nano formulation because the colloidal particles will be able to go into the tumor because of leaky vasculature. So that's also the enhanced permeation and retention effect. What's happened? Good permeation, retention. Why retention? Your lymphatic system is poor. From the tumor, the lymphatic system is very poor. So that whatever goes, it's not very easily cleared. So that's how it's enhanced permeation and retention effect is there in the passive targeting. Active targeting, I already mentioned. Your active targeting can be always in nanotechnology. It can be also antibody drug conjugates. Your drug and antibody can be conjugated. Why antibody? Because antibody is found to be having a receptor antigen present in the particular tumor. The tumor will be having a receptor pattern, like EGFR, receptor receptor, like tyrosine kinase receptor. So that means you are actually conjugating your drug, or you're conjugating, decorating your nanoparticle surface with a molecular antibody and the ligand, anything which is able to identify the tumor cell which is present in the body. So what is triggered release? Triggered release is nothing but you design the drug such that you design the complex such that it only releases in certain conditions. What conditions? It can be to release, it has to produce a heat, it may have to produce a magnetic field, or it can also be a low pH. If you take the tumor has a acidic pH. Acidic pH will be promoting the drug release. That means your body has a pH of 7.4, but whereas the tumor tissue has a acidic pH, maybe 4 or 5, I'm not exactly sure what was the pH. So that means drug is released in the acidic pH better. That means healthy tissues drug is not released, only drug is released in the acidic pH. That's how tumor targeting is done. So basically what we have to do in the cancer targeting is you'll have hundreds of patients. If you don't do the molecular profiling, why you have to do the molecular profiling? Every cancer type, every person, what generally happening most of the cases is any person who is diagnosed with the cancer, we are just giving a chemotherapy of cyclophosphamide, the cisplatin, the doxorubicin, the taxas. These are the drugs we are giving it. We are not really understanding which drug may be effective. How will we be able to understand which drug will be effective? You have to understand molecular pattern, which receptors, which proteins, which type of the tumor type it is, which drug may be effective. If you do the molecular profiling, that kind of personalized therapy can be done. That means you have to segregate the patients into based on the molecular patterns, what kind of the markers they are carrying on, so that accordingly I can select the doctors and we will be able to better give the right drug. The breast cancer person having a certain type of receptor, if you are giving another type of drug, that means not going to be effective, and so on, right? So that's why you have to properly pattern the molecules for either disease type or the cancer type or at the molecular level. Then since once you know the molecular level, you know the particular the molecular patterns, you have certain drug. You can actually give the drug so that you can completely avoid the safety issues caused by that, as well as see the efficaciness. You're actually unnecessarily not dumping the drug into the patient. So that's how you are going to get a very good response. This is what the dream is, but this is not nowhere we are close to this, and that's where you need to have more the molecular tools in the cancer. As I mentioned, antibody drug conjugate and cancer targeted therapy is something very promising. Why this is very promising is, why antibody I'm talking about antibody which is found to be having a antigen present in the tumor cell. Anytime you talk about the antibody has an antigen present in the tumor cell. It can be receptor, it can be enzyme, whatever it is, a cellular component, a cell membrane component. That means you are trying to make a recognition of the recognition to the tumor cells, and what you are doing, you are conjugating the drug with the antibody. So if you don't conjugate the antibody, what happens? Drug will be going everywhere. The moment you conjugate the antibody, now my drug the distribution or targeting to the tissue is properly controlled. How it's controlled? Because more accumulation can be expected when we have drug actually going to the drug is driven by the antibody. That's why you can use a different surface receptors, antigens present on this, right? These are the antigens. If you see the table, CD30, CD24, all these particular type of receptors are found to be present in the cancer cells. When the moment you have this knowledge on this, you can actually develop the monoclonal antibody against this particular antigens. Monoclonal antibodies are nothing but biologics. Then you conjugate the antibody with the drug. Then you can actually reduce the toxicity issues. So that you can also reduce the dose also, right? If you know that whatever the dose I give, you only go to the tumor, you can also reduce the dose. Instead of 100 mg or 500 mg, maybe you will be needing 100 mg or 50 mg. So significant dose reduction is also possible, right? So you will be using a linker to do that. Linker will be having a property to get broken when they go and bind to the receptor. So if they have to be antibody and drug conjugates, will be having a linker to deliver it. So this is what is the concept stuff. So once it go to only to the tumor tissue, it is going to selectively kill the cancer cells by sparing the healthy tissue. Another concept here is the CAR therapy, right? CAR therapy is chimeric antigen receptors. Basically, this is more of an immune therapy. Immune therapy in the sense you try to produce the immune cells, the cells which are going to, you are actually taking the chimeric antigen receptors. Basically means you are expressing a receptor on the cell. What cells you are taking? You are taking the T cells. T cells, if you take them from the blood, you will have T cells. So when the person is diagnosed with the cancer, so when we have a CAR therapy possible in your establishment hospital, what they do is they take the patient bloods. When they take the patient blood cells, they isolate the T lymphocytes. The T cells can be isolated. When you take the T cells, now T cells, the T cells are actually immune cells. Now what I can do is I will engineer my T cells such that I am going to express one receptor which is called CAR, chimeric antigen receptors. So what I am doing is I am trying to express a receptor on the T cells, and I will ask the T cells to proliferate in the body, in the platelets. I take the T cells, I use the gene editing tools or the gene knocking tools to express the receptor on the T cells, and I'll grow these T cells in the petri plate, expand them after the gene is inserted. Once the receptors are present on the T cells, now I will try to inject these T cells, same T cells, which are modified T cells to the patient. So when I inject back to the patient, what happens is, since my T cells are modified to having a receptor, the receptor is specific to the cancer cell. That's called CAR therapy. So you can able to, that's called the living drugs. It is living drugs. Why? Because the cells are living and they're acting like a drug. There is no drug there. It's just a T cell which is acting like a drug. And you'll expect no safety issues, right? But is it available in the India? No, it's not available. It's a very expensive tool. But imagine if your body itself is able to attack the cancer cells because of the CAR therapy, the kind of safety we can enjoy is going to be huge. That's another advantage. So somebody want to read more on the CAR therapy, you should read it, and it's very interesting. And I'm just giving you the idea how the CAR therapy works, but there's a lot to go on. There is also PROTAC based targeted therapy. What is PROTAC? Proteolysis targeting chimera. Again, here also there is a one target, like c-MYC. MYC is oncogenic target is MYC, MIC. MYC is an oncogen. Now I will not able to directly target that. So undruggable targets. c-MYC is not a druggable target. So there is a technique called the proteolysis targeting and targeting chimera, degrading the undruggable protein, so that you have a PROTAC approach. Through that, you can able to target the undruggable approach by using this trick. So what you will be having in this is, one example is ARB825 is an PROTAC molecule composed of two ligands. We have two ligands. One ligand will be targeting a undruggable one. Other one is acting like a ubiquitin ligase one. So through this, what you can do, what I'm trying to say here is, there's a PROTAC approach not just for the c-MYC, it can be also for other targets as well. So again, here the knowledge of the molecular biologist, knowledge of the chemistry is required, because you have to design certain linkers, spacers, and ligands are required. So let me quickly jump to the cancer stem cells. Despite of having all these different approaches, we're still not able to get the good cancer therapy. You have molecular therapy, you have a PROTAC therapy, you have so on. Because these all the therapies are only targeting one cancer type, one type of cancer cells. The problem is your tumor tissue is not just one cell type, it is a heterogeneous population. One of the concept is CSCs. CSCs are nothing but cancer stem cells. Cancer stem cells are something, stem cells are having a self-renovation, right? So what happens is, if you give the chemotherapy, 99% of the tumor is shrinking. Only 1% the cells are not dying. These cells are nothing but the stem cells. And the stem cells, since they can regrow again, they will give the tumor relapse can be very much expected. This is what exactly going to happen if cancer stem cells are present in the tumor. Because you are giving a doxorubicin, you are giving some molecular therapy, targeting 99% of the tumor cells have that particular receptor, but the 1% of the cells have the cancer stem cells, they are very resistant to all kind of chemotherapies, all kind of targeted therapies. That's where you also have to target the cancer stem cells. That's why you have to design a drug, you have to have an approach where 99% killed by the conventional therapy, as well as 1% death. 1% of the cancer stem cells also needs to be targeted, so that we'll be able to reduce the complete eradication of the tumors is possible, as well as there is no relapse can be expected. So traditional, if you have a cancer stem cell, as I mentioned, only 1% is a cancer stem cell. Traditional cancer therapy, which is only remove the bulk of the tumor, you still have a stem cells present. If you only target the stem cells, the bulk of the tumor still can go in. The best approach should be combination therapy, where we have a conventional cancer cells, normal cancer cells are killed, as well as the cancer stem cells are also killed. If you see, we have taken from this cell in 2015. That means a lot of approaches, cancer stem cells can be targeted to completely to better target the cancer basically. If this speaker is talking about how cancer stem cells can be targeted, so a lot of molecules, lot of receptors, lot of targets, if you see. And another important aspect of the cancer stem cells is they have a very highly overexpressed efflux pumps, ABC transporters, right? The highly overexpressed ABC transporters like ABCG2, like P glycoprotein, they have multi-drug resistant pumps. That's why, so we there are different ways. Either you inhibit the efflux pumps, or you actually try to inhibit the one of the molecular patterns which are found to be involved in the cancer stem cell survival. So different ways you can actually target it. WNT ligands, beta catenin pathway, and the gamma secretase inhibitors, SMO antagonist, hedgehog signaling, and so on. The cancer stem cell is such an area you will enjoy reading it. Just some of you, if whoever is keen to know it, just read about cancer stem cells. Very interesting topic it is. If you take the strategies and therapies targeting cancer stem cells, the surface markers and signaling cascades, microenvironment, epigenetic mechanisms, other strategies. So different strategies are there where either you're using the CD receptors, you're having signaling cascades, or you're actually talking about microenvironment like chemokine receptors, or vascular endothelial growth factors, or weakly acidic pH, histone distillate, like so on. And as I mentioned, other strategies like metformin. So metformin is an anti-diabetic drug. Came here. How it has came here? Because metformin has a different mechanisms. So metformin, people are trying to repurpose it because it's a well-proven drug. If we can actually, metformin may not be directly anti-cancer, but if metformin is making your tumor tissue kind of conditioned such that it is now more feasible for me to give the chemotherapy. So better targeting can be possible. So that's how the metformin in people are, so like anything. So if you think the nano formulations are tried with the different cancer stem cells like glioblastoma, breast and colon cancer, and these are the mechanism involved in that. So this is the table is talking about, right? So the different types of nano formulations, people are developing it where they use the conventional therapy as well as the therapy which is also targeting the cancer stem cells so that we can actually able to better control it. So how do you target the tumor cells? How do you use nanotechnology to deliver? What is the targeted therapy? I mentioned it has to be an antigen drug antibody conjugate. It can be a nanoparticle which is surface conjugated with a ligand. What are the examples? You have folate receptor overexpressed, you have transferrin receptors overexpressed, you have mucin receptors overexpressed, you have the YS peptide which is selective EPH2 receptor, you have IRGG protein which is found to be highly effective in promoting the tumor distribution, and a CD44 ligand for the cancer stem cells, and so on. Like you have targeting can be done by one of the few ligands at the surface receptors present in the tumor cell, or we can also identify the new receptor which is maybe identified through the omics or other tools, right? So when we did one of the experiment, this was during my postdoctorate, when I was trying to target the EPH2 receptors. EPH2 receptors actually are tyrosine kinase receptors which are found to be overexpressed in breast cancer. When we give the docetaxel through the nanolipid carrier, where we have conjugated the NLC with YSA peptide which is binding to the EPH receptor, we can see a very significant increase in the tumor drug concentration of the docetaxel. Only 4,000 nanogram concentration, we can see more than close to 12,000 nanogram concentration in the tumor. That means that the amount of the drug goes to the tumor is highly increased when we have done in the targeted therapies. We have used YSA peptide during my postdoc. So that's possible, right? So you can increase the tumor distribution. Similarly, we have multifunctional nanoparticles. Multifunction means one nanoparticle have multiple functions. Functions can be happening on the surface. Functions can also happening inside the nanoparticle. Why inside the nanoparticle? Instead of one drug, you can take two drugs. One is two drugs targeting different pathways. One may be targeting the stem cells, other may be targeting about mixed population cells, whatever it is. Or one can be drug, other can be a diagnostic agent, or a diagnostic agent. Or surface may be decorated with not just one, it may be 2-3 different type of moieties. Why? Your tumor again cell is also having a heterogeneous, right? One type of tumor person, same cancer type, one patient may be having a one type of receptors more, other person may be having another type of receptors more. If you have a group of targeting ligands on the nanoparticle surface, if one receptor is not present there, another ligand will be driving the drug to nanoparticle to go there. So multifunctions are always good, and very good to as a theory, but if you have to develop the multifunction nanoparticles, it's a huge task, because the moment you optimize one, other may not be properly fitting in, right? So you have to optimize multiple things: size, surface chemistry, drug loading, amount of the and stability of the ligands which are adding it. So everything matters. That's why multifunctions are complex formulations, complex nano formulations. But if that someone can develop that one, it is going to be a game changer, right? So there are a lot of advantages, as mentioned. The multifunctional nanoparticles are not going deeper into that. So multifunctional nanoparticle, if you are planning to do that, you have to have the properties, stability and biocompatibility. You can use polyethylene glycol and so many other things. Surface specific targeting, you can use any of the antibodies, peptides, or aptamers. Intracellular penetration, you can use the peptides. Imaging, you will be using it in quantum dots or the magnetic nanoparticles. Stimulus, sensitive drug release, will be using a pH or photosensitivity and so on, right? So not necessarily you have to incorporate all these the properties. Some of the properties which you think are going to increase the performance of the drug. You know that now the nanoparticles already promoting the safety. Means drug chemotherapy safety concerns can be reduced by the nanotechnology. Now if you can use further improvement through the targeting or the multifunctional, you may be able to get the efficacy as well as improve. So multifunctional example is like that. You can use different ligands, imaging agents, gene therapy can be possible, different type of chemotherapies can be done, stealthing effect to promote the circulation of light, and different delivery vehicles can be tried. Multifunctional nanoparticles can be done. Different nano systems like liposomes, polymeric, metals can also be tried, like gold or other nanoparticles people are trying. So this is just a model figure where it's well covered about how the and remember it's so complex, but none of the industry or the science working the group is working on trying to incorporate all. They only pick one from each property and they try to optimize it, right? Now, so we talked about the cancer therapy, we talked about the CAR therapy, we talked about so many. But however, one concern is still there. What are maybe your drug? How good may be your targeted strategy? Shiv Shankar, how much time you have? Shiv Shankar.
Sir.
How much time we have?
Sir, you can carry on, sir.
So, when is the next speaker joining?
We have up to 11, 15, 11:30.
Okay, so I have a lot of slides, but I think I'll by 11:15 I will complete it. Okay, 11:10 I will complete it. Right? Is that fine? I will complete by 11:10. Right?
Oh, okay, sir. No problem.
Okay. Whatever may be the strategy, whatever may be the approach you are trying to work on, there is one concern. Left side figure is talking about chemotherapy before anti-fibrotic agents. Right side is talking about after anti-fibrotic agent. What I am saying is, if you remember, I was talking about the passive targeting. Passive targeting means since your tumor has a leaky vasculature, drug will be easily escaping from the blood vessel and goes to the tumor. It just crosses the blood vessel barrier, but from there, the drug or the nanoparticle to penetrate further not possible. Why it's not possible is, if you might have touched some of the tumors, I have touched in the animal tumors, they are very hard. Why they are very hard? Because they are highly fibrous. Highly dense extracellular matrix is present. What is happening with the extracellular matrix, dense extracellular matrix? Increased intratumoral pressure, ITP, intratumoral pressure is increased. Increased hydrostatic pressure in the tumors. Due to that, Though nanoparticles reach the escape from the blood vessel, it's called extravasation. From there, reaching to the all parts of the tumors is not possible because of the high fibrosis nature. Due to the same fibrotic nature, tumors are highly metastatic in general. The more fibrous the tumor is, more collagen and the more extracellular matrix in the tumor is, the more metastatic it's going to be. Means it can actually, the matrix will promote the tumor to go to other locations. So generally, that's the belief. Not only metastasis, it is also preventing the distribution of the drug to the distal part of the tumor, right? So if that is the case, there is a way, there are the strategies people are trying to do where we can use anti-fibrotic agents. If you use anti-fibrotic agents, fibrosis is nothing but extracellular matrix, right? So if you can cut down the extracellular matrix like collagen through certain approaches, like we have anti-fibrotic agents, they may be able to promote the better distribution. What are the compartments which are not reaching the drug earlier because of the fibrosis can be minimized by using an anti-fibrotic therapy, so that drug will be distributed through the tumor. So no part of the tumor is not having a drug. So that's how, as you know, that if one part of the 1% of the tumor is not getting the drug because of the fibrosis, we will have relapse. The relapse can be prevented here. That's why anti-fibrotic therapy can be effective, so that better distribution. This is my experimental evidences. Left side, if this is the control, I'm saying there is a blue color, right? Blue color is indicating, blue color staining is indicating that if you see there is kind of round circles are forming. I'm showing with the red arrow, right? So what they're showing is there are the collagen bundles are forming around the tumor tissue. This is a lung tumor tissues which I have done in my postdoc. What's happening? The extracellular matrix is making a compartment. That means making a round wall around the particular tissue. What will happen tomorrow if you don't, if the drug, so this round of the compartmentalization, intratumoral compartmentalization which is taking place here, may not be allowing the drug to penetrate inside inside the compartment. Then what will happen? Rest of the tumor might have got the drug, but that while inside the compartment, the drug is not reaching there. Due to that, what will happen? You will not able to see any drug penetration, so that the tissue is still intact, and the tumor will again go much better than virulent than the previous one, because it might have got limited amount of the drug. So like that, right? So that means intratumoral compartmentation is seen in the tumors, in the animal experiment. Same is also the case in human tumors. That's why. So you need to make sure that drug distribution is properly taking place. That's why we have tried losartan, telmisartan. Why losartan, telmisartan? And there is a clinical trial done in the MIT with losartan, where they are giving the losartan, priming up losartan before chemotherapy was promoting the better distribution of the drug into the tumors. That's one approach there. Then we compare the losartan, telmisartan was better anti-fibrotic agent, because they are all both are inhibiting the TGF-beta. TGF-beta is a male mineral in the fibrosis. So we are trying to inhibit the TGF-beta to the FDA-approved drugs like losartan, telmisartan, these are anti-hypertensive drugs which are having a well tolerated. Then followed by either chemotherapy, maybe. So if you see the right hand, losartan, telmisartan, we don't see a lot of collagen deposition. That means the collagen accumulation is lost. Collagen accumulation lost means the compartmentalization has been removed. That's what is happening there. This is the hypothesis we have made there. The left side figure, if you see the thick blue color, the violet color, the bundles are there in the tumor. That means the thick sheets are there. That's why nanoparticles or the drugs are not properly penetrated. When we give the anti-fibrotic agents like telmisartan, losartan, we are seeing a very uniform distribution of the nanoparticle into the tumor. That's what we have proven through the fluorescent microscopy. What is happening here? In figure A, we don't see a lot of red fluorescence, whereas the B and C, because of the increased distribution of the fluorescent nanoparticles, like polystyrene nanoparticles, we could see increased distribution. So we have taken fluorescent nanoparticles as a model nanoparticles. Tomorrow you can take one of the FDA, like toxil and the ambroxil. They all will be able to better distribute. That's what we are saying is, You give the antibiotic therapy initially, you can able to better promote the distribution of the drug through the tissue, tumor tissue, so that relapse and the better eradication is possible. This is one approach, right? If you see this figure on the left side, this is how you must appreciate this. There are collagen bundles which are blue in color are very well formed in the tumor tissue, so they will not be allowing the drug to go inside the compartments. This is exactly the concept. In relation to telmisartan enhances intratumoral distribution of nanoparticles in lung cancer model. This is the paper which I have published in the Journal of Control Release. This is the recent paper, it is 11 point something. I published this work in 2030 in my postdoctorate. So this is the concept we have done it. We have given inhalation. Why we have given inhalation? Because inhalation is easy for the drug to go into the lung. Right? That's why we have given inhalation delivery of telmisartan, increase the intratumoral distribution of the nanoparticles. That's what we are talking. Another thing is there is enzyme called lysyl oxidase. Lysyl oxidase, LOX2, not lipoxygenase, it's lysyl oxidase. It's the enzyme is very vital for the collagen to become a monomers to polymers. Collagen polymerization is promoted by lysyl oxidase. That's what we have identified. So what happens is, whenever there is a tumor is becoming very hard, that's because of the lysyl oxidase increased lysyl oxidase activation, which is promoting the collagen monomers to become bundles, collagen bundles. These bundles are making the compartments. That means there is a scope for us to inhibit the lysyl oxidase to the chemical inhibitors of the any other tools. We have tried to develop the siRNA or shRNA as a tool to inhibit understand whether lysyl oxidase targeting can reduce the tumor fibrosis as well as promote the distribution of the nanoparticle. When we knock down the lysyl oxidase to the ssRNA technology, which is where we actually selectively knock down the lysyl oxidase through the ssRNA, we could see the relevant markers are decreased. TGF beta, which I mentioned, is decreased. And when I see the control TNBC and the treated, what is TNBC? Triple negative breast cancer. Fluorescence is limited in the control, whereas in the last two treated cells, the fluorescence is high. Why is the fluorescence high? Because I have knocked down the lysyl oxidase too. My tumor tissue has a better penetration to the nanoparticles. So when we do the collagen specific staining, very huge staining is happening in the TNBC control in the panel B, whereas limited collagen is taking place. What we are saying is, last two knockdown actually is promoting the distribution of the nanoparticles because it is removing the collagen fibers. So followed by we also given the how the chemotherapy the doxil. As I mentioned, doxil is what? Doxil is a doxorubicin liposomal formulation is called doxil. As I mentioned, but doxil also promoting the better safety profile, but efficacy is not well proved and improved compared to the doxorubicin. The reason is distribution is not taking place. Due to that reason, what we have done, we have initially given the LOX2 to the animal. After LOX2 siRNA is given to and then we have knocked down the LOX2 enzyme. Then when we given the chemotherapy, doxil alone and the doxil followed by LOX2 followed by doxil, we could see the last animal tumor is almost disappeared, right? Compared to the other, this is the breast cancer model, right? So what we are saying is tumor volumes and the tumor burden is significantly reduced when we have knocked down the LOX2 followed by given the doxil, which is a doxorubicin liposomal formulation. The take-home message here is what it is that any approach, any pathway you target, you say that I am trying to reduce certain phenomena. Here I am talking about the tumor fibrosis, or I am talking about the cancer stem cells earlier, right? Since distribution is not proper, I am almost going to the last part of it. Since tumors are not promoting the distribution of the free drug as well as the
challenge evaluation could 10,000 rupees
free drug as well as the nano formulations. So you can actually think of tumor penetrating peptides.
Minimum 5,000.
That's where recently, not recently, almost 10 years back, there is a concept called IRGD peptide. I stands for integrating peptide. That means the RGD, which is promoting the integration of the nanoparticles into the tumor, also called the tumor penetrating peptide (TPP).
amount.
So right, so IICTE is a tumor penetrating peptide. It was discovered by Rosalotti et al. And there are 2 Indian scientists involved with this. No, they were actually IICT scientists. Now they actually have their own companies in the US. So they are actually members in this particular activity. What they have done was that phase display assay. Phase display assay tells how the protein is binding to the certain target. So when through the phase display assay they found that IICTE is a peptide which is forming to the tumor. That means binding to the tumor tissue. Based on that, they found that alpha 5 beta 3 and alpha 5 beta 5 as well as neurophilic receptor overlaps in the tumor, and this IICTE peptide works with that. What's happening here when you give the IICTE peptide injected to the tumor bearing animal and the human, it is becoming C and R rule. C and R rule is actually the it's a compulsory. It's a C and R rule. C having a C end which is having a arginine moiety. So the C and R role is forming it. That is required for this particular the arginine peptide to bind to the neurophilic receptors. These neurophilic receptors are found to be overexpressed in the tumor vasculature. Now, due to this IICTE peptide cleavage to the proteolysis, now this the IICTE cyclic peptide is becoming a chain peptide and having a C end, having a R region. That's why I call the C and R rule. And this will be surprisingly promoting a very, very effective distribution of the drug into the tumors. Why I'm saying is, I'm still surprised how this is promoting it. Can you imagine? If we give the drug together, you give the IICTE peptide plus a free drug or the nano formulation, no conjugation, you just inject physical mixture. This IICTE peptide is promoting a increased distribution of the unconjugated drug into the tumor as well as the conjugated drug into the tumor. That means just have to inject the IICTE peptide plus your drug, what are the drug you are talking about, what are the chemotherapy you are talking about, and they are actually promoting the distribution of the drug to the tumor. We can conjugate it, we can also no need to conjugate it, both can be done. So this is what the evidence is. That means IICTE peptide is one of the very, very effective peptide. There is a lot of literature, there is already company formed on that I think they are doing the clinical trials on this. I'm sure if this is proven clinically, this is going to be a very fantastic achievement because a peptide is found to be safer and it is promoting the distribution of the drug to the tumor tissue very selectively. This is what exactly the examples they are doing in that paper. This is not my work, it's a work taken from science paper, right? So what's happening here? Left side, free doxorubicin IICTE, free doxorubicin. Red fluorescence indicating that how much is the distribution is taking place in the tumor. So if you see in the free doxorubicin, not much, only a few spots. Whereas in the left side, everywhere you have the red fluorescence. What's happening? Free doxorubicin plus IICTE, just a combination, no connection, no conjugation, just a physical mixture. And we see that doxorubicin accumulation in the different organs. You can see a tumor tissue has a almost 7-fold increased accumulation of the doxorubicin, which is very significant, right? From 1 microgram, one unit, it had gone to 7 units, 7-fold increase in the accumulation of the drug. Whereas in the other healthy organs, it was found to be decreased. That means, so that means what's happening is your tumor is really promoting your IICTE is promoting the doxorubicin free drug to go to the tumor, better distribution. Another example is the doxorubicin liposomes, that's called the dox plus IICTE. Why IICTE? Again, it's a physical mixture. Plus means just a combination they are given. Here only with the doxoliposomes. So what they have done is, since doxorubicin can give the red fluorescence, you can see there is no red fluorescence happening in the just doxorubicin liposomes. Whereas if we give the IICTE in combination, the distribution is increased. That itself is indicating, right? Whatever the drug, the free drug or the liposomes, you can improve the penetration of the drug into the tumor tissues through the IICTE peptide, which is going to be very, very unique. Another example is ABX. ABX is nothing but Ambraxane. Ambraxane is the first nano formulation approved by US FDA for cancer therapy. Ambraxane is a NAP technology, nanoparticle albumin bound technology, where albumin nanoparticles were prepared by loading paclitaxel into like albumin. So since albumin has a lot of albumin receptors overexpressed in the tumor tissue, when they did that, abraxane alone is not distributing. Green fluorescence is decreased. Whereas abraxane is given in conjugation or in combination without conjugation. In both the cases, you can see a distribution is improving. This is going to be very, very unique in my opinion. I have no words to explain this. How you know? Without a conjugation, the IID is promoting. Conjugation, you understand. You have a link which is connected. They will go together. But you know, if you use the combination, they are also still promoting it. So these papers have all come the cell and nature. So we cannot even tell. They have done a lot of repetitions as well. So that means among all the different targeting approaches, if we can do this IRGD, if we can do more exploration, there can be potential benefit we can expect for to treat the cancer patients. Basically, so you can use nanotechnology as the targeted therapy to cancer diagnosis, to deliver the drug selected to the tumor. You can also homing in the sense, deliver selected to the tumor tissue, so that better imaging, better localized therapy, and selective killing of the cancer cells can be possible. That means a person who goes to the clinic with the cancer, by identifying the right approach, if we can diagnose and treat the localized therapy or the targeted therapy, we'll be able to completely eradicate the tumor. That's the dream actually people are trying to have it, right? I think I'll stop it here. Though I have a siRNA, shRNA, messenger RNA, and microRNA, this is another area where for last 5 to 10 years, microRNA has become a very important topic in pharmacology and drug delivery, because microRNA is found to be having a role in almost all the diseases. In certain cases, it's increasing. MicroRNA is not just one; it's a group of microRNAs, miRNAs. Why microRNAs are so important is, unlike siRNA or shRNA, microRNAs can control 100 different messenger RNAs. That means if I can identify the right microRNA, I can able to control the messenger RNAs which are involved in that particular disease condition. So one microRNA, I can able to control hundreds of messenger RNAs. That's the beauty of it. So I think those who are interested to know more on this, you can learn on this as well. So I think I'm stopping it here. I hope actually I made some impact on you. So I didn't go too much deeper into that, but I just try to give you a light on that, so that you can go back and read it, right? So everything is available. Just have to go to the right article and find the right source to read about it, right? So there's a lot of ways where you can actually enjoy your science by reading also, right? With this, let me thank all the participants for your patient listening. Very sorry in case if I have actually repeated some of the content which you already might have known. Thank you so much for all the opportunity. Thank you, guys.
Sir, thank you very much, sir.
Yeah. Let us have questions if you have any.
Yes, sir. We are expecting any questions from the audience or from the participants.
రేష్మా మన దాంట్లో అప్లోడ్ చేశాను రిజల్ట్ అనాలిసిస్ ఒకసారి తీసుకొని వెరిఫై చేసి బాడీకి మార్చుకోవడమా ఆటోమేషన్ ఉంది చూడండి అప్లోడ్ చేసాను మార్క్స్
Any?
Sir, myself, Dr. Sheikh Munir, sir.
Okay, can you keep your video on? Okay.
Sir, thank you very much, sir, for delivering your topic on this promising trends in the pharma sciences. Sir, I have a small questionnaire, sir, related with the multifunctional nanoparticles.
Yeah.
You have stated about the importance of this multifunctional nanoparticles. Why? Because as of with respect to this role of nanoparticle in terms of COVID, usually you had explained with the amphotericin B. So where it is producing the nephrotoxicity.
Yes.
So where further, sir, in order to give or in order to reduce this nephrotoxicity, you have said about this ampicone.
Yes.
So now, sir, is there any marketed formulation available with this multifunctional nanoparticle as of now?
Whatever the knowledge I have, I think there is nothing is approved so far. And because as I mentioned, it's very complex,
Yes.
very complex system there. No, you cannot develop everything. Maybe 2 functions together, 3 functions together. The concept wise, it's people are aware of. Now, the only thing is that now we are not able to just make the liposomes, ambosomes in the laboratory scale. You see, doing at laboratory level is different. Doing at the clinical level, translatable level is different. So I think there are no formulations, if I'm right. Maybe some clinical trials might be happening. I didn't see the clinical status. Maybe, I think, as of now, I think there are no formulations so far available, in my opinion.
Thank you, sir.
Yeah.
And thank you very much, sir, again. And we also discussed about a role of omics in the drug discovery. And Shiv Shankar, sir, any questions, sir?
Sir, here you used.
Yeah.
Sir, these anti-fibrotic agents in chemotherapy, will they pose a threat like metastasis? Something can happen if you are using this anti-fibrotic agents, because is there possibility of these cells, cancer cells, migrating to other places? Metastasis, will there be a problem?
Actually, yes, wonderful. Exactly, this was the same question we have expected. We got from when we submitted a grant to NIH through my postdoc. This concern needs to be also kept in mind. But you are giving an followed by you are giving chemotherapy, right? Followed by you are giving chemotherapy right away. So you're not giving a lot of window, right? So you should be able to target. But there is a concern. It can be increasing the metastasis. But there are a lot of clinical trials happening in the MIT Harvard. There's an Indian group called Rakesh Jain group is working on this. Maybe I think that needs to be that angle also needs to be seen. But since you are giving a chemotherapy followed by that, even if it is going to other organs also, chemotherapy may be killing it.
Thank you, sir. Thank you very much.
So participants, no questions. Only faculty there. So it's a formality you are asking. no participants. I could see 37 or 45 people have joined. Maybe they might be observing from the YouTube.
From here. Thank you again, sir. Thank you very much for your valuable presentation, sir.
I hope I hope I at least I conveyed some of the trends, if not the only ones. I hope I have covered some content which will be useful to the participants. Thank you. Yeah, please carry on.
Now, sir, we request you, sir, to accept our felicitation on behalf of. Sir, we request you, sir, to accept our virtual felicitation, sir.
Okay.
In behalf of our SR.
Somebody is trying to raise the hand. Is this something? Go ahead. Otherwise, it's up to you. We can do that later. Yeah, we carry on, sir. Sorry. We carry on, sir. Thank you. Thank you very much. Thank you. Nice.
Sir, thank you very much.
Thank you.
Now, I take it a privilege to invite our next speaker, sir, Mr. L.A. Ramprasad Garu, Director, JLS Health Sciences Private Limited, Hyderabad. Very good morning, sir, and we invite you for this Impact Lecture Series Session 2. Now I request Mr. Madan Gopal, sir, to give a brief biodata about Mr. L.A. Ramprasad Garu. Madan, sir, please.
Thank you, sir. Good morning to one and all. This is Madan Gopal, Associate Professor, Department of Pharmacy, Santhiram College of Pharmacy. It's my immense pleasure to speak. Pleasure to introduce our second speaker, who is going to talk about the process of establishing a startup. Is none other than Dr. L.A. Ramprasad Garu. Coming to the academics and a professional category for the L.A. Ramprasad Garu. Sir completed his B Pharmacy, Andhra University, Vizag in the year 2000 and later he's completed his post graduation in the subject in the department of pharmaceutical and food analysis in the year 2002, and his PhD degree in the Andhra University, Vizag. And coming to the professional, are having a more than 20 plus years of industrial experience in this in the field of research and development, in which more than 15 years in a leadership and decision making role. And sir now working as a director, JSL Health Sciences Private Limited. It is a startup company for the pharmaceutical formulation, CRO. Previously, sir worked as a DGM in Hetero Lab, Hyderabad, and later assistant manager, analytical development department in Cadila Pharma, Ahmedabad. Then junior manager, generics, Dr. Reddy's Laboratory, Hyderabad. Coming to the key responsibilities, key capabilities for this one, sir has ability to plan, develop, and manage analytical support for pharmaceutical development, pharmaceutical development from pre-formulation through the launch process in a multidisciplinary team environment. And sir having a experience in set up a AD laboratory with CGMP documentation and implementation of quality systems as per the regulatory market requirements. And also he has sir having a handful experience in the analysis of various dosage forms and responding to the regulatory queries pertaining to the analytical issues for various regulatory markets. Coming to the major contribution by the sir, he oversaw the analytical functions and provided value added analytical support for Hetero's first NDA approval and other P4, NCE 1 and IND projects, and played a pivotal role in project initiation, strategy building and development, batch execution, and ANDA submission, response to queries and approval. Coming to the academic achievements by the sir, sir received more number of prizes for poster presentations we attended in various conferences conducted across the nation. Coming to the training, sir attended more number of training programs like Milford, USA, Shimaju, Asia Pacific, Singapore, and the dissolution testing conducted at advanced dissolution testing conducted at Mumbai. And also sir presented his research work in national as well as international conferences. This is about the regarding profile about the Dr. L.A. Ramprasad Gar, sir. Thank you, sir.
Yeah, thank you very much for a very nice me.
Madan sir, mute yourself.
I consider myself as a very fortunate 2 years. Yeah, good morning, Professor Garu.
Carry on, sir. Please carry on, sir.
Yeah, just a brief introduction about our company, startup company. I have chosen this topic is last year we have started a new formulation CRO. So I thought that this topic, when Professor Shivashankar Garu told me like which topic you would like to opt, then and he has given me a choice. Then right away I have selected this topic because starting a startup and having an idea of starting a startup is good, but when you start implementing, when you try to really start this seriously, you will be facing a lot of problems. First thing is your your friends, relatives, and so there will be a lot of discouraging things. There will be a lot of encouraging things. You know something, and you think what you know, maybe you're really not knowing it. So there will be a lot of confusions while entering into entrepreneurship. So last one and a half year, I faced a lot of these things, and I felt I lacked some of the guidance while initiating this startup. Okay, so I thought this would be a right platform to guide youngsters like you
and people who are attending this will be having a better idea of initiating a startup or starting their own business, particularly in pharma. If you Google, you will be having number of much of information. But thing is, how well you are going to utilize it for successful entrepreneurship. So that's the reason why I have chosen this topic. So to start my presentation, a brief presentation of mine. Just a minute. Will you be able to see my presentation?
Sir, not yet, sir. Not yet.
Yeah. Just a second. Yeah, is it okay now?
Yes, sir.
Yeah. So our company name is JSL Health Sciences Private Limited. Basically, it's a
Namaste, sir.
formulation CRO. Yeah.
Can you put it in the presentation mode, sir? PPT is in editable mode.
Okay. Okay. Just a second.
Sir, presentation mode. Maybe I think you have to share entire window and then select the PPT.
I have done that only. Just.
Sir, otherwise only select the tab and share, sir.
Yeah. Is it okay now?
Sir, 1 minute. Sir, it is in not yet, sir.
Yeah, again, yeah. Listen, yeah.
Sir, share through a window mode, sir.
Yeah, just a second.
Sir, please.
I think you received it, right?
Yes, sir. Yes, sir. We have received it.
Okay. Yeah, thank you. So, shall I continue? Yeah. So, am I audible to you? Yeah. Thank you. So, our company is JSL Health Sciences. It's a startup pharma companies with our services. Like we do formulation development of solid orals, liquid orals, dry powders, suspensions, injectables, ophthalmics, complex ophthalmics, 505 B2 kind of formulations, and NDDS. May I go to next slide, please? So it was my dream. Like since my starting of the career, I was having a idea of starting my own company. So so a group of our friends collaborated. I mean, we came together and we started all our experience. Like we have a cumulative experience of more than 150 years. All are technical experts in their fields. They are from various fields in pharma industry, like from formulation, from analytical, from regulatory affairs. So many they contribute all their experience in starting our company. See, the key differentiator of our company strategy is, if you see, it's design, develop, and deliver. So before starting our company, we have done a small pilot survey. So what is the current industry practice? How is the current CRO business in India and abroad? And what are the gaps? And how best we can fill up those gaps. So there were gaps between the CROs and the industry. That means the customer and the client. So we wanted to bridge those gaps. So until unless you have a strong design, until unless you have a strong knowledge of what you are going to develop and deliver, and if you doesn't have any strategy in developing that, definitely you will be ending in a mess. So you have to have a design in your hand. So before design, you have to have a strategy. So how to design and how well you can deliver it. So all these things you have to put it on a paper so that you can develop in a systematic way. At the same time, you can develop a robust formulation. Then delivery. So how well you can, how excellently you can, deliver the product without any hiccups during the execution at plant level. So all these three components are very much important for a CRO company. So within 1 year gap, we have set up. May I go to the next slide, please? So in a 1 year setup, as I told, like we have done a pilot survey, we have designed our business model, then we have registered our company at the Ministry of Corporate Affairs. Then we have set up. We were working in various other companies, so we disengaged from those companies, and we have started working on independently. We rented one office, and we started working on our facilities. We started funding, crowdfunding, and getting the funds into that, because it was a huge expense, like more than 5.5 crores. So we have already invested. So that much funding we were not able to do it on ourselves. So we went for a crowdfunding, and we pulled up a lot of money, and we started. So and then we applied for approval from the Pollution Control Board, CFE and CFO. Almost we reached more than 40 clients. We have already written and signed up more than six MOUs with major CROs, as well as CDA agreements already done with 4 multinational companies, and few more under discussion with some abroad companies. Soon we will be getting into an agreement with them, and we will be working on some new INDs, new investigation new drugs. So the next few slides are a brief note of our services. So may I go to next slide, please? Yeah, these are all. Next few slides are brief introduction of our services. The most important part, what I would like to say is this: I want to participation of few of the audience, like how many of you want to start a company? Because the topic of today's session is process of starting a company, a startup. How many of you would like to start their company? Yeah. So I guess a lot of people will be definitely interested in starting their own company, because it's a value creation thing. Just if you go to the next, if you skip next 4 or 5 slides, I request to move on to slide number 10. Yeah, value addition, value creation, being a role model, and it could be a dream of someone who wants to have their own company, right? So value addition, if you are working for some company, and your presence should make a value addition. It's not like 9 to 5 job or 10 to 6 kind of a thing, but your presence should make a difference. At the same time, you should add a value to it. So the company, whoever employs you, should feel that yes, this person or this fellow is an asset for me. So that one should do for their company if they are working. If you want to really create a value, then start a company. So value addition is yes tough, and value creation is much tougher than value addition, right? So when you create a value with ethical business, then you will be a role model. When you become a role model, yes, then you can show your dreams to others. Say everybody will be having their own dreams, but being retiring in a responsible position or respectable position, or reaching a high level in your career, all these things dream. But how can you show your dreams to others? You can explain, but you cannot show your dreams to others. Unless you create those dreams, then only people will understand what you have dreamt a few years back. Okay. Then, may I request the next slide, please? Yeah. Paperwork. So when you are going to start a company, so you have to do some paperwork. So what kind of a paperwork is? The slide number 11. Yeah. Yeah. So distinguish among the different types of and sources of startup ideas. Because we are in pharma company, there is a lot pharma companies or pharma industry is a ocean, right? There are a lot of options starting a business. So you have to distinguish between the different options, different startup ideas, different sources. Be it a trading, API trading, be it reference standard manufacturing, be it intermediates or pharma excipients or formulations, distribution. So many things are there. Similarly, regarding services, regulatory services, development services, clinical services, pharmacovigilance services, there are so many services. So you have to distinguish between the various sources, various ideas. To excel in entrepreneurship, you have to have a innovative thinking. So these are all existing one. So in future there will be lot more options like artificial intelligence in drug delivery, artificial intelligence with respect to precision medicine, personalized medicines, right? So you have to have a novel thinking. So put it on a paper. So if you have an idea, put it on a paper and check the feasibility. Then you have to analyze your business model. Like what are the external factors which will be influencing your business financially as well as the commercially. At the same time, internal analysis, what kind of capabilities you have to run the business. Then think broad-based strategy options. So if you are finalized with the business option, business model, then you start thinking in a broad-based. Like broad-based means if you want to start up a pharmaceutical CRO like us, so what are the various options available globally? Who are going to be your clients? So what you need to have in your hand so that you can start and run a business, right? So all these things, business options, time that is going to from startup to ending with successful orders. So all these things you have to think and analyze so that you are mentally as well as physically and prepared for the all the circumstances, like having business in hand. Without business also you need to wait for some time and until you get business in your hand, right? So for all those things you have to mentally as well as the financially be prepared for that. Otherwise, sometimes you'll find yourself in a big jungle. Definitely you will get depressed. So we faced all these things and we came a long way in last 1.5 years. So before starting this, we had a lot of brainstorming. Before 2020, we started in 2016 with this idea, and it took more than 4 years to really start a startup. Okay, once all these things done, assess the feasibilities. Feasibility with respect to, you have to have a good facility. You have to have a list, like what you need to have in your facility, right? What are the technical manpower you need to pull in your venture, so that they will be supporting with their intellectual inputs as well as their technical knowledge. So all these things have to be weighed in a proper way. Then only you have to start your business. If you are not doing anything, any plan, then it's very, very risky. So if you are failing to plan, then you are planning to fail. So do it before entering into this field. Yes, there will be some hardships, there will be definitely some challenges, but once you enter, you should not see anything other than you have to focus only on your business. Why I'm saying this, I have seen businesses, I have seen my friends who started business and really they felt very, very, tough, and their they're withdrawn from their business ideas. They sold all their businesses because they haven't done any paperwork previously. So once you are in, and you have to see the success. Then only you should not, get depressed. You should not get discouraged. My humble request: if you want to really start a business, be strong. Yes, there will be hardships definitely, but be strong. Everything will get resolved when you have patience. Can we move to next slide, please? So as I said earlier, like what you want to manufacture, what you want to deliver in your business, whether it is a product or a service. So if it is a product, what kind of a product? Yes, if you want to manufacture at a prototype or if you want to manufacture at a bigger level. So all these things matters. Yes, you have started if you wanted to manufacture a product, whether is it really beneficial to the public or not, whether it is catering the needs of the public or not, whether you are adding a value to the consumer or not, whether it is affordable or not. If you are manufacturing an unimportant thing, there are so many similar kind of products in the market. If you want to add 90th product, similar product, no one is going to purchase it, right? Similarly, whether it is a hatke kind of a thing, like it's entirely a different kind of a thing, and or though it is a smaller one, if it is having a huge benefits. Yes, you can. And at the same time, if it is whether it is affordable or not. That means if you want to sell your product at 100 rupees per piece, you should get it manufactured below 40 or 50 rupees. So you have to do it all these things before entering into the boxing ring. Then opportunity recognition. So identification of potential new products or services that may lead to promising business. See, you may ask a question, like because you have already started a CRO, there are n number of CROs, then why you have started already a similar kind of a business or similar kind of a service? As I said, we have done a pilot survey. There is a gap between these kind of CROs and the industry. So what industry is expecting from CROs and what existing CROs delivering to the industry, when we assessed it, there was a gap. So we entered into this business to bridge that gap, to capture those kind of opportunities where these gaps are there. So that kind of a thing you need to do. Then entrepreneurial alertness. So sometimes opportunities, I have seen people losing business because they have not identified those opportunities. Sometimes opportunities will clearly visible. Sometimes there might be a hidden opportunity. So few things you need to do, anticipating a bigger opportunity behind that. So the strategic moves as well as the alertness to identify those opportunities behind a hardship one should have. So that will give you a blockbuster which makes you a crorepati overnight. So you have to be alert, and at the same time you should not be greedy to grab all the opportunities. So strategic moves, identifying or anticipating a big opportunities behind and each and every step is important thing one should have who wants to become an entrepreneur. Let's move on to next slide. So the types of ideas into startups, right? A new market or a new segment or a new technology or a new benefit. So before starting a company, so you have to identify your strong point. You have identified a new market, new population, new consumer group, or you might be having a new technology in your hand which can be commercialized, which can be increased, scaled up, or a new benefit for the existing product. So which one you are having in your kitty? Accordingly, act accordingly. So that would give you a faster success.
అదే ఇవన్నీ చదివిన తర్వాత ఇవి కట్టడం అంటే లాస్ట్ ఉన్నాయి సార్ ఇవి ఏమన్నా టెంప్లెట్లు ఏమన్నా మాకు
నెక్స్ట్ స్లైడ్ ప్లీజ్.
నేను చూసాను సార్ ఇది ఫస్ట్ ఇది ఒరిజినల్ ఆర్టికల్ ఇది చూసాను.
So what happens is
ఈ రెండు చూసాను.
most of the businesses are started based on prior work experience like us, because you will be having a hands-on experience, how to operate, when to enter, what to do, what not to do.
అంటే మీరు నిన్న నేను ప్రిపేర్ చేసి పెట్టాను సార్, మీరు చెప్తాను.
And sometimes personal interest as a hobby, or some of the businesses like wedding planning hobby, or some multi level marketing, it's a hobby for some people. Some people enters a business development service because they want to go around the world. So and their career is a chance to gives a chance them chance to move around the globe while marketing. Sometimes by chance they enter. For sometimes if you see those statistics, so most of the things happen, most of the businesses are started by people who had already a working experience on that domain. So having experience definitely adds to your business. If you are starting a business on which you hadn't have any experience previously, that would be really a risky step. Unless it is a very common business, unless it is a family business. Yeah, using innovative thinking to generate business ideas. So how to generate? First of all, you have to have an idea, great idea, good, strong work experience, in-depth knowledge of any idea, any business model, because starting from the scratch and turning it into a successful, when people recognize as yes, he has become, when people start you identifying as this, he is a successful business person, then that is the journey towards that level is needed. When you have a correct idea and correct action and correct focus. So borrow ideas from existing products or services, some other working on that business. You may combine 2 or 3 businesses. Suppose if you are manufacturing and you are having a distributor, you can start distribution also. Or if you are having a distribution chain, to have a big margins, you can start manufacturing. Or you have a distribution, you doesn't have a marketing, advertising, you can start advertising. So combine, not only, don't rely only on one business, try to combine 2 or 3 businesses like vertical integration or backward integration kind of a thing. That will give you good experience as well as the capturing as much opportunities as possible. For example, we wanted to start a formation CRO. But we had an opportunity to supply APIs to some of the countries. Because I was having a good network in API with API manufacturers, I grabbed that opportunity, accepted the order, and we started trading with APIs. So that helped in generating some business, some revenue, which helped our business. Similarly, you can start with others. For example, another example, if you are having a distribution of diagnostic kits, then you can start with a diagnostic center. You can consume your own diagnostic kits. At the same time, if you add some more capital, you can purchase some diagnostic machines like CT scanner or MRI scan. So you can add some more revenue, you can diversify. Just like in the share market, you have to diversify your investments. Similarly in business also, you have to diversify your capital and try to grab as much opportunities as possible. And identify what is the hot trend. Last 2 years we have seen COVID. So I have seen people who made a lot of money during COVID season, and I have seen people who lost all of their money. So there was a lot of demand for sanitizers, masks, gloves. People who imported gloves from Malaysia, they minted money. After some time they lost again. It's all like there are so many businesses. Thing is, you have to identify what is the trend, what is the idea, how to do, when to do, what to do. People who started this kind of businesses in second wave or late, they lost their money. People who anticipated earlier in the first wave itself, they made money. So you have to be very alert in identifying the trends and explore ways to improve an existing product or services function. As I told previously, already I told, like you have to identify the way of improvisation, whether it is a product or be it a service. If you are manufacturing a hand sanitizer, add a soothing component into it, which is hands friendly. Because earlier we used to rub. People have complained with burning sensation on their hands. And people who started adding aloe vera gel into hand sanitizers, they started advertising hands friendly hand sanitizer. It gives a soothing effect. So a different marketing strategy, different advertising strategy. And once customer enters into your facility, or once customer adds to your list, you have to retain that customer. So retain all your existing customers and add new customers. And customer retention activities and customer problems, addressing customers' problems or needs. So all these things you have to have a separate team who takes care of your customers. Customer service, all the businesses, like be it a telecom department, be it a DTH, whatever it may be, everyone have their separate customer service departments, right? How well you serve the customer, that many times of customers you will be adding to your business. So customer addition, customer retention, and customer service is a it's a big department. So bringing a customer and at the same time retaining retaining your customer, existing customer is also very much important. For example, global pharmaceutical market up to financial year 2024, it will be increasing. Post-COVID, it has increased and it will increase. So this created a lot of business opportunities. So what are those business opportunities? Everywhere there is an increase in expenses. If you see previous consumption of dry fruits or nutraceutical products has increased multiple times post-COVID. Instead of depending on only medicines, people have realized that the importance of having a good food in their dinner plate or in their lunch plate. So that is the reason now you have seen number of dry fruit shops. It's also a business. It's also a part of healthcare industry. So now pharma industry, not only a pharma industry, it is like a healthcare industry. Personal hygiene is a big business. Healthy food, organic food, nuts and fruits, squashes, fruit juices without preservatives. Even the packaging industry, though it's an ancillary packaging industry. So preserving good food, preserving pharmaceutical product, preserving anything which is consumed by the public. So packaging industry, you have to have a very good appearance of your package, right? So impressive packaging aesthetic should be good so that it can attract the consumers. So now the pharma industry has grown into other fields, other ancillary fields. So altogether it's a healthcare industry. Yeah, next slide talks about various statistics of various other countries. Their spendings on healthcare and pre-COVID, post-COVID scenarios of this altogether healthcare industry and their spending over health. Yeah, next slide please. Yeah. So you have to assess the competitive environment. So you have an idea. Now you have checked the feasibility of those ideas. You have assessed your business model, created your facility. You have identified gaps. You have entered into your customer's place. Customer came to you. You have a customer service department separately. But that is not the end. After you, again there is a chain of entrepreneurs who already entered or waiting to enter. So you have to assess your competitor. While playing cricket, nowadays there are a lot of statistics, sports statistics. You have to assess your competitor's strengths and weaknesses. Once you enter, you are going to be a part of some others' pilot survey. You will be surveyed by somebody else. Your business model, your way of work culture, your way of your strategies, your products, your delivery systems, everything will be assessed by some other to develop their business strategy. So, what equipment you have to have is, before they enter, you have to assess the next generation's ideas. What my next person will be doing it to enter into the business, to grab my business. So you have to keep an eye on your followers. If you have already grown to a level, no competition will affect you, then it's good. When you are entering, when you have just entered, keeping an eye on your competitors and competitors' strengths, their weaknesses, their customer base is also a very much important step while starting your own business. Yeah, next slide please. So evaluating market opportunities. So, as I told, there will be a lot of opportunities. You have to knock the right door. So, evaluating an opportunity is also a big quality that one should have, one entrepreneur should have. Every opportunity may not give you money. Some opportunities will give you name, some opportunities will give you money. Similarly, some opportunities will make you lose some money, some opportunities will depress you. But still, it's an opportunity. If you get name and money, then it's good. If you get depressed, if you get a failure, if you lost some money, don't worry, it's a lesson. Try to learn a lesson from your failure and do not repeat it. That is one of the key ingredient of whole, this idea of starting a business. And at the same time, you need to have a very good team who have a competitive potential. Surround yourself with similar mentalities, similar psychology, similar potential, similar ideology, so that everyone will be contributing their ideas, their strength, running your business successfully. Second, having resources in your hand. Even manpower is one of your resource. Your equipment is resource, right? So all these resources, how to get best resource into your facility. Try to acquire those resources. So where to go? Where you will find these kind of resources at a best price and best quality. And yeah, and your capability. Sometimes what happens is your business will be so big that you will be incapable of handling that. Then you have to have a good team who can handle all those services. I mean, all those divisions. So, having capability of running a business, at the same time having right people in your right people at right places. Just like cricket, you have to put a right batsman at right position, right? Who is going to open? Who is going to be a tail ender? So put right people at right places, right? Then what are the core competencies of yourself? Who is heading the business? At the same time, core competencies of your team. That is going to be your core competency or core potential of whole your firm. So all these things are parts of evaluating marketing opportunities. So you have to evaluate business options, you have to evaluate people in your team, you have to evaluate your customer, you have to evaluate your total business model. So if you are doing a business today, and what are the growth drivers for the future? If you are doing it today, if you are not sure of what's going to happen tomorrow, then it's a failure. So you have to have a roadmap for the next 10 years or 15 years. If you want to do a business for today, tomorrow, this weekend, then I will start, then again I will start after 5 years, after 5 months, 4 months. No. So when you are starting a business, if you want to stay there for a long period, then you have to assess the growth drivers of next coming 5 years. Post 2017, there was a hidden slump, recession kind of a thing in pharma industry. There was a situation when few people Their businesses, pharma industry is on peak. Yes, there were some challenges. Yes, accepted. But people who took their challenges into their stride, they really, made some money. They got a huge success. So, next 10 or 15 years, you have to have the growth drivers in your hand. If you have identified good growth drivers, then what is your action plan to meet that growth? So every year you have to change your working style, your strategy to meet that growth. So whenever you are doing business, you will be anticipating it is a exponential growth, right? So to have that, you have to expect more from the coming years. What is going to be the hottest? Hottest means a good product in next coming 4 or 5 years. For example, in pharma industry, what are the patents that is going to be expired in coming 3 or 4 years or 5 years? Most of the people will be working on next coming 4 or 5 years. But you have to work on patents that is going to expire or patent in non-infringing or circumvention things that is posing a challenge for general pharma companies till next 10-15 years. There are companies which are working on patents which are going to expire in 2038, almost 16 years from here, but still they are working as on date. Why is? If they can circumvent, if they can bypass that patent, they will be having a good success, huge success. So you should have a mixed basket which will give you immediate revenue next 3-4 years, mid-term goal and short-term goal, mid-term goal and long-term goal. So you have to work simultaneously on the products or services from all these 3 baskets, so that they will give you immediate revenue, mid-term and for long-term. Work on a product where there is a very tough patent is there, try to non-infringe it, circumvent it, file it, get the approval and demand a royalty which is 10% or 7% or 6% for next coming 15 years, 20 years or 30 years. That is your long-term goal. Short-term goal, immediately next year there is a patent expiry, you want to launch a generic, yes, start it. You have a technology ready, do it and launch it. That is immediate money. If you are working on a product which is going to expire, whose patent is going to expire in 2025 or 2027, yes, it is going to be your mid-term goal. Multiple people will be there in mid-term goals. Short-term goals, there will be less competition, and long-term goals, there will be less competition. Most of the competition will be in mid-term goals because it's a overlapping area. Yeah. May I go to next slide, please? So once all these things are done, you have to assess, you have to do a SWOT analysis. Your strengths, your weaknesses, opportunities and threats. So having strengths, opportunities is good. But most of the people won't accept or won't focus on their weaknesses. If I started a business, I know what are our weaknesses, and we will be continuously working on that to improve those, to reduce the number of our weaknesses, negative factors. At the same time, simultaneously put an eye on the threats, market threats. For example, hospitality industry never expected this corona, so they suffered a lot. So of course, it's an inevitable situation. Nothing was there in anybody's hand. some things which will not in our hand, we cannot do anything. But thing is, you should be ready. It's a lesson for us. Corona is a lesson. So you have to have acumen to anticipate those negative impacts which is going to affect your business. So do all these things, then continuously work hard to improve your strengths. Yes, there will be you will be never reaching a position where there is no negatives or no weakness or no threat. Only thing is you have to improve your strengths and opportunities areas so that you can outweigh your weaknesses and threats. Threats will always be there, weaknesses will always be there. No team is stronger. Be it Australia, England, only thing is you have to make your team such a strong level so that you will not be losing any match or without fight. So you have to have strengths in you so that you can outweigh your weaknesses. Grab every opportunity so that you can reduce your threat. Yeah, may I go to next slide, please? Yeah. So, in brief, market factors, competitive advantage of your business, how well you can attract your consumers, your customer base, how well you can manage your capabilities, how best you can reach your consumers, and how well you can minimize your fatal flaws. So one wrong step may pull you into a big pool. So do all this work before entering into a business. There will be some hidden challenges, there will be some visible challenges. Yes, visible challenges you can address. Hidden you cannot unless you face it. So when you do all these things, sometimes the ground will be much deeper than what you have anticipated. Only thing is you have to balance. So balance between your commercial thoughts, your capabilities, then followed by your business. So collaboration. Why collaboration is a key integral? It may bring you some capital. It may give you access to new technologies. And is collaboration simply, it may help you to overcoming the barriers. For example, a big pharma company may acquire you. A big company much bigger than you may come and acquire you. That will help you, go into regulated markets. Or you may get the access to using a high-end technologies. Recently there was an acquisition. The small company which was working on a relatively new technology on a prototype, they have just submitted a patent application. A very big pharma company have noticed that these people are working on so and so product, and they have really done inroads into that patent. Definitely that patent, if granted, is going to be a very big commercial success. Immediately they acquired that company for an unimagined price. That business value of that company previous 2 years back was around 20 to 30 crores per annum. It was projected to 50 crores coming to next 2 years. And this company was bought almost 460 crores. That business volume was around 50 crores when projected to coming 2 to 3 years, and that was bought for a value of 460 crores. It can happen with any business. Thing is, if you have right business idea, if you are working on a right service, right product, which serves for, which is going to be stay in the market, in the industry for years together, coming next 10, 15 years, yes, then success is in your stride. Definitely you are going to get a success. No one, nothing can stop you. Definitely you will get it. Then the way forward. For example, we all belong to pharma industry. So if you are working on something, you have to have a way forward for this. Because every 5 years, I have seen for the last 22 years, I have seen pharma industry taking a turn for every 4 to 5 years. Because pharma industry is a evergreen industry. Now it has become a healthcare industry. Every 5 years there is a path-breaking technology or a path-breaking, new dosage form kind of a thing, new therapeutic region, immediate lot of demand for one therapeutic category, oncology, lot of demand, lot of new drugs entered, lot of new dosage forms entered, lot of new nanoparticle, liposomal drug delivery, MABs, monoclonal antibodies, and for existing diseases, lifestyle diseases, for diabetes, there is an artificial intelligence controlled insulin delivery system. You have to stick a patch onto your body. There will be artificial intelligence chip. It will be continuously assessing your blood glucose level. If your blood glucose level is higher than the stipulated limit, it will trigger insulin to deliver into your body. If it comes to normal level, again it will start. See to what extent the technology has gone is everything will be monitored, and all these things, all this artificial intelligence, drug delivery, the chip working, assessing, giving result, that triggers a indication that again delivers the drug, again it stops, again it starts. All these things will be monitored by a doctor who is sitting in another city or in another country, or if you are traveling to another country, everything will be monitored from India. Your near and dear, your relatives can see. So what's happening with my dad? What's happening with my mom? So whether they are taking medicine or not, not to worry. Everything will be, that chip will be doing. Only thing is you need to put that patch on your body. So technology is developing. Thing is you have to utilize it right way and at the right time. Having a business idea and taking it to the commercial level, this journey is very, very tough. Only thing is you have to have a very good discipline. Unless you have a discipline, by what time you are waking up in the morning, what kind of exercise you are doing, because you have to keep your mind fresh and very alert to do all these multiple tasks, assessing your customer, assessing your competitor, assessing your business, assessing your product. Every day is a challenge. Unless you are mentally fit and strong, physically strong, you will not be doing all these things. So before starting a thing is, you have to do all this paperwork. Do lot of paperwork on this, then only enter. Why I am telling all these things? See, registering at NCA, getting a GST number, getting a PAN number, getting a CFA approval, getting a CFA approval. All these things, you can Google it and you can learn, you can get things. If you hire a consultant, everything he will do, he will get ISO certificate, he will get that approval, he will get this approval, everything he will get. Thing is, developing the idea to implementation and value creation, your strategy building, your team build up, all these things, nowhere it will be written. And only your experience will tell you. That's what I am doing today. So, thank you very much for your patience listening to my lecture. I tried to make it brief. I think this is a brief thing. This is what I wanted to tell you, all the attendees. So very soon we are going to have a very big partnerships with some of the big pharma companies. And I wish all success to all the participants. Thank you.
Sir, thank you very much, sir. We expect any questions from the participant. Thank you very much, sir. Is there any participant? Are there any questions from the participants? Now I request Suresh sir to speak few words, sir.
So, thank you, Ram Prasad Garu, for giving the session, information giving regarding the establishment of startup. And at the same time, I also thank all the delegates for participating in the seminar. And at the same time, I will expect your cooperation in the future seminars also. Thank you very much, sir. Thank you. Munir.
Definitely, thank you. I love to guide or give some lectures or knowledge, share my knowledge, what I have gained in my career. And we encouraging a lot of young students to take over their project work in our facility, and most of the times we make them doing experiments. It's not like just listening, coming, reading SOPs kind of a thing. We encourage them to do practically also. Most of the, I don't think most of the pharma companies will allow to do things. I allow them to do and learn, and sometimes I ask them questions. Whenever I enter into the laboratory, I ask what they are doing, what they have understood. Because until unless we have industry institution interface, that these students will not learn. When they connect this academics as well as the practical, then only they will they can do miracles. That's
Yes.
what I believe.
Yes, sir. Correct.
Yes.
Correct.
Now we request Ram Prasad Garu to accept our virtual felicitations, sir, on behalf of Santhiram College of Pharmacy.
Thank you. Thank you.
So now I request Dr. Shivashankar Reddy sir to present vote of thanks, sir.
Good afternoon to everyone. I, Dr. Shivashankar Reddy, professor working at Santhiram College of Pharmacy, would like to propose vote of thanks on this Institution's Innovation Council Impact Lecture Session 2. The theme of the lecture is Entrepreneurship and Innovation. My sincere thanks to Dr. Up startups along with strengths and pitfalls and the precautions to be taken while setting up of a startup. Thank you very much, sir. I also thank our principal, sir, for guiding us to conduct this program. My heartfelt thanks to Dr. Munir, Dr. Pradeep, Dr. S.V. Suresh Kumar, sir, Mr. Maheshwar Reddy, Mr. Madan Gopal, Mr. Niranjan, and all the professors and co-faculty for extending their help in successful completion of this impact lecture. I also thank you all, the participants. With this, we come to an end of this Impacts lecture session 2. Thank you one and all. Jai Hind. Back to Munir.
Thank you very much, sir.
Thank you all.
So really very thankful, sir, both for the Chandray Garu and Ram Prasad Garu for your valuable presentation on this impact lecture series. So which is a key aspect for the student so as to have the interaction with the industry on the current prospects. Thank you very much, sir. And I thank all the participants or delegates who have shown their interest to participate in this AICTE sponsored Impact Lecture Series Session 2. And thank you very much to the management and the principal, sir, and all the fellow faculty members. Thank you very much, sir, to making this a grand success. Thank you, sir.
Thank you. Thank you. Nice meeting you, Dr. Ram Prasad Garu.
Yeah, thank you. Thank you. Actually, I worked on Ebrexel, Paclitaxel, NAPAC.
I'm sure.
Also.
Nice. Okay.
Previously, I worked on few Lipurlite, Doxorubicin, Paclitaxel.
Where did you work on this, sir? Which company?
Hetero.
Okay. So, do you have the product Abraxane from Hetero now?
Generics, yes, but domestic market they have.
Okay.
Now they are working on regulated markets also.
Oh, nice. Because there is a lot of scarcity for this nano formulations.
Yes.
One product is successful, I think you will have a lot of business. One formulation is enough, I guess. Like Doxil, Natco was enjoying like anything.
Yes. The thing is like having a prototype is okay, but during commercialization, continuous production, there is a lot of challenges.
That's what I was also telling the participants. A proof of concept is fine, but when you have to translate for the large scale, you will face a lot of problems.
Exactly.
Yeah. Exactly. And nice, sir. Where is your company located?
It is in Kukatpalli, Prashant Nagar.
Oh, okay. We are nearby then. We should meet once.
Exactly. Yeah. Correct.
Yeah.
I will visit you once.
To take your number and details. I will meet you, sir, one day. We'll see how we can work together.
Yeah.
Thank you.
I hope, sir, last request to both the speakers. As we are in academics, we will come to you and we request you to give us some projects which we can collaboratively do. And of course, we may not live up to your expectations, but with your guidance, we can learn from you a lot and do some publication together or some sort, sir.
No, Sivashankar Garu, please don't say that we may not live up to your expectations. Definitely, you can do miracles. We respect academic institutions a lot. People who identified that potential, they succeeded. That is for sure. Who neglected, ignored academic institutions, they failed. Or they will fail. Definitely, academic institutions have that potential. Only thing is, I should not say, but lack of funds, they are, maybe to some extent, it is one of the drawback. There is nothing less than any industry, this one. Definitely.
I totally agree with you, sir. See, it's not that only certain people can do it. It's
Yes.
So you have gone through the courses, You are the masters and PhD in that area, so I'm sure you will be able to contribute it. So I always try. No, actually, I kept on slide also. What kind of research can be done? Actually, since the lack of time, I could not go through it. So I think you have to pick up the area where you are capable. It means feasible for you. Not to think of very complex science where you'll end up disappointment because you'll not have a lot of stuff to achieve that. So there is a lot of scope. To be frank, you can address a small problem that can give you a lot of
In US and Europe, industry encourages institutes, colleges. There are lots of collaborative research work is happening in US and Europe.
Correct.
So we should encourage it.
Sir,
I think we have one AICTE project funding of around 16 lakhs, which have been recently released in the year 2020 and 21. I think I should request Chandraya sir, because we have put the theme in which you are working through. So
okay.
I think we will meet again somehow in the next coming 1 or 2 months, sir.
Definitely.
Your appointment will come over there and we'll surely discuss about this.
No problem. So wherever we get an opportunity, wherever we think we can actually help you, designing or see the progress of your projects as hypothesis. We are there, and we don't think so. NIPER are only institutes can the big labs can do it. Anybody can do the science. It's all you need to be smart and you need to identify the right problem at the right time.
Okay.
That's all is required.
Yes.
Okay, sir. Thank you very much, sir, for
Thank you.
your kind cooperation and accepting our invitation. Thank you, sir.
Thank you. Bye.
Bye.
Bye, sir.